Hello there, and welcome to this quiet space where the deep past can unfold without hurry. Tonight, we're stepping back, not centuries or millennia, but hundreds of millions of years, to a moment in Earth's history when the very nature of life changed. This is not a story of sudden drama, despite what the name suggests. The Cambrian Explosion, as scientists call it, was explosive only when measured against the slow clock of geological time. It lasted perhaps 20 million years, a span almost impossible to hold in the mind, yet brief enough that paleontologists still debate what made it happen so quickly. Before this window opened, Earth's oceans were quiet. For tens of millions of years, life existed in forms we would barely recognize as alive. Soft, passive organisms anchored to seafloors covered in bacterial mats. No mouths, no eyes, no movement, just existence, slow and still. Then something shifted. The planet crossed a threshold. Within that 20 million year window, nearly every fundamental body plan that defines animal life today appeared in the fossil record. Creatures with skeletons, with eyes, with the ability to hunt and flee. The blueprints for all subsequent complexity, laid down in a geological instant. What caused it? That's the question we'll walk through tonight, step by careful step. Not with urgency, but with the patience the subject deserves. We'll trace the accumulation of oxygen in the atmosphere, the flood of minerals into ancient seas, the genetic innovations that allowed bodies to assemble in new ways. We'll follow the first predators and the arms race they triggered. We'll look at the destruction of the old world and the construction of the new, and we'll do it all at the pace of continental drift, letting each piece settle before moving to the next. If you're somewhere comfortable, that's good. Let your breathing slow. The story ahead is long, and there's no need to rush. Before we begin, I'd be grateful if you'd let me know where in the world you're listening from tonight, and whether the sky outside is light or dark. It's a small thing, but it reminds me that these questions, ancient as they are, still gather us together across distance and time. If this kind of patient exploration is something you'd like more of, a quiet tap on the like button or subscribe helps this reach other curious minds. No pressure, just an invitation. So settle in. We're going back more than 500 million years, to the moment when complexity became possible, then inevitable, then permanent. Let's begin. Every animal you've ever seen, every creature with a backbone, every vertebrate that has walked or swum or flown across this planet, traces its lineage back to a single narrow window in deep time. 540 million years ago, give or take a few million years, something fundamental changed in Earth's oceans. The change was not instantaneous, not in any human sense, but when measured against the 3 billion years of microbial life that preceded it, the shift was startlingly abrupt. Before this moment, life was simple. Bacteria, archaea, single-celled organisms floating in vast seas or clustered in thin films on the ocean floor. Eventually, multicellular forms appeared, soft-bodied and passive, shapes that look alien to modern eyes. Then, in what geologists call the Cambrian period, complexity exploded. Not literally, of course, but the metaphor captures the speed and scale of the transformation. Within roughly 20 million years, the fundamental architectural plans for animal bodies, the phyla that still define biological diversity today, appeared in the fossil record. This is the inheritance we carry. The human body, with its bilateral symmetry, its centralized nervous system, its tube-like gut running from mouth to anus, is built on a blueprint established during the Cambrian. So is the body of every fish, every insect, every worm. The diversity we see around us, staggering as it is, represents variations on themes composed more than 500 million years ago. No fundamentally new body plans have emerged since. Evolution has elaborated, refined, adapted, but the basic architecture was set during that brief geological window. Understanding why this happened, why complexity arose when it did and not before or after, requires tracing a convergence of planetary conditions. The Cambrian explosion was not a single event, but a cascade, a sequence of interlocking changes in chemistry, climate and ecology that together created the circumstances for complexity to emerge and persist. It was a threshold crossed, a tipping point reached, after which the planet could not return to its previous state. The story begins not with the explosion itself, but with the world that preceded it, a world so different from our own that it requires careful imagination to grasp. For tens of millions of years before the Cambrian, Earth's oceans were populated by organisms that did not move, did not hunt, did not flee. They simply existed, absorbing what the water provided, anchored to mats of bacteria that carpeted the seafloor. This was the Ediacaran period, named after the Ediacara Hills in Australia, where fossils of these strange organisms were first discovered. It was a world of stillness, a world where the rules that govern modern ecosystems, predation, competition, rapid adaptation, did not yet apply. To understand the Cambrian, we must first understand what came before. We must step back into that quiet world, the last era before movement and predation transformed life on Earth. The Ediacaran biota, as these organisms are collectively known, represent an experiment in multicellular life that ultimately failed. They were evolutionary dead ends, displaced by the more active, more complex forms that emerged in the Cambrian. But their existence and their eventual extinction is essential to the story of how complexity began. The Cambrian Explosion did not happen in isolation. It was the culmination of processes that had been building for hundreds of millions of years, processes that slowly, incrementally, prepared the planet for a transformation it could not reverse. Oxygen accumulated in the atmosphere, nutrients washed into the oceans, genetic systems evolved that allowed rapid morphological change, and when all these pieces finally aligned, the threshold was crossed. What we see in the fossil record from that time is not chaos, but the sudden appearance of order, of structure, of bodies built to do specific things, bodies with hard skeletons for protection, with jointed limbs for movement, with eyes for detecting threats and prey, bodies that could burrow into sediment, disrupting the microbial mats that earlier life depended on, bodies that could hunt, creating the first sustained arms race in Earth's history. This was not just biological change, it was ecological upheaval, the replacement of one planetary system with another. Every subsequent expansion of life, every later diversification event, has occurred within the constraints established during the Cambrian. The Great Ordovician biodiversification event, which followed roughly 50 million years later, saw an explosion in the number of species, but not in the number of fundamental body plans. Evolution has been refining and elaborating the Cambrian toolkit ever since, but it has not invented new toolkits. The Cambrian closed a door behind itself. It established the rules for what animal bodies could be, and those rules have held for 500 million years. This is the world we inherit, a world where complexity is the norm, where predation drives adaptation, where movement and sensation and internal specialization define what it means to be an animal. But it was not always this way. There was a before, and understanding that before, understanding the quiet, passive world of the Ediacaran, and the slow accumulation of conditions that made change possible, is where the story must begin. When scientists speak of the Cambrian Explosion, they are describing an interval that spanned roughly 20 million years, from 541 million years ago to approximately 530 million years ago. In human terms, this is incomprehensibly long. In geological terms, it is a blink. To understand why paleontologists consider this rapid, it helps to situate it within the broader timeline of life on Earth. Life itself is ancient. The oldest confirmed microbial fossils date back nearly 3.5 billion years. For most of that immense span, life remained simple. Bacteria and archaea dominated, single-celled organisms that reproduced by dividing, that metabolized energy from sunlight or chemical reactions, but that never developed the internal complexity we associate with animals. Around 2 billion years ago, a crucial innovation occurred, the eukaryotic cell, with its nucleus and specialized internal compartments. This was a necessary precursor to multicellular life, but even after eukaryotes appeared, the transition to large, complex organisms took over 1 billion more years. The first multicellular organisms, the Ediacaran biota, appeared around 575 million years ago. They persisted for roughly 35 million years, a respectable duration by any measure. Then, at the boundary between the Ediacaran and Cambrian periods, something changed. Within the following 20 million years, a span that represents less than 0.5% of life's total history, most of the major animal phyla appeared in the fossil record. Arthropods, mollusks, echinoderms, chordates, the lineage that would eventually give rise to vertebrates, all emerged during this narrow window. This is what scientists mean by explosive. Not that it happened overnight, but that the rate of morphological innovation, the appearance of fundamentally new body plans, occurred at a pace that has never been matched since. It is as though evolution, having spent billions of years experimenting with cellular life, suddenly discovered how to build animals and then tried out every possible variation in rapid succession. Measuring this event requires precision. The start of the Cambrian period is formally defined by a specific marker in the rock record, the first appearance of a particular trace fossil called Treptichnus pedum, a burrow made by an unknown organism. This marker represents the moment when complex mobile life began to actively disturb the seafloor, leaving behind evidence of its movement. The choice of this marker is not arbitrary. It reflects a real ecological shift, the transition from a world where life existed on the surface of sediment to a world where life could move through it. The end of the explosion is harder to define precisely, because it was not a sudden stop but a gradual settling into a new normal. By the Middle Cambrian, around 510 million years ago, the pace of new body plan, innovation had slowed. The fundamental architectures had been established. Subsequent evolution would elaborate on these themes, adding species diversity and ecological specialization, but the underlying blueprints remained stable. One of the challenges in studying the Cambrian explosion is that the fossil record from this time is incomplete. Soft-bodied organisms rarely fossilize, and even when they do, the preservation is often poor. Most of what we know about early Cambrian diversity comes from a few exceptional sites, places where unique geological conditions allowed soft tissues to be preserved in exquisite detail. Without these sites, our understanding of the explosion would be far more limited, biased toward the minority of organisms that evolved hard shells or skeletons. The most famous of these sites is the Burgess Shale in Canada, which preserves Middle Cambrian fauna in remarkable detail. But the Burgess Shale dates from after the initial explosion. For understanding the earliest phase of diversification, the Chengjiang biota in China is more important. Chengjiang captures early Cambrian life, documenting the presence of complex body plans, including the earliest known protovertebrates, within just a few million years of the Cambrian's start. This finding was crucial. It confirmed that the diversification was not only rapid, but front-loaded, with much of the morphological innovation occurring early in the period. The speed of the Cambrian explosion raises a fundamental question. Why did it happen so quickly? Evolution does not suddenly accelerate for no reason. The rate of morphological change is constrained by genetics, by development, by the availability of ecological niches. Something must have changed to allow or compel such rapid diversification. The answer, as we will see, lies not in a single cause, but in a convergence of factors, geological, chemical, and biological, that together created a unique moment in Earth's history. The Cambrian explosion was not simply the appearance of new species, it was the invention of new ways of being an organism. Before the Cambrian, life was passive, absorbing nutrients from the environment, anchored in place or drifting. After the Cambrian, life was active, mobile, predatory, prey. Bodies became tools for survival in an environment where survival was no longer guaranteed. The pace of change was driven by the urgency of this new reality. Once predation emerged, every organism faced a choice: adapt or disappear. The arms race had begun, and it has not slowed since. This chapter has outlined the temporal boundaries of the explosion, the 20 million years during which complexity became established. But understanding when it happened is only the first step. The deeper question, the one that will occupy the rest of this exploration, is why? What conditions had to be met for the explosion to occur? What changes in the planet's chemistry, in its climate, in the genetic systems of early organisms made this possible? We turn now to the world that preceded the Cambrian, to the quiet, passive ecosystems of the Ediacaran, to understand what had to change for complexity to emerge. Before the noise and motion of the Cambrian, there was quiet. For tens of millions of years, Earth's oceans were populated by organisms that existed in a state of profound passivity. They did not hunt, they did not flee. They simply were, anchored to the seafloor or lying flat against it, absorbing what nutrients the water provided. This was the Ediacaran period, spanning from roughly 575 million to 541 million years ago, and the organisms that defined it, collectively known as the Ediacaran biota, represent Earth's first sustained experiment with large, multicellular life. The Ediacaran world was fundamentally different from the one that followed. Imagine an ocean floor carpeted with soft-bodied forms, some resembling fronds, others like quilted mattresses or disc-shaped impressions. Most were anchored in place, their bodies oriented to maximize surface area for nutrient absorption. They lack the features we associate with animals. No mouths, no guts, no eyes, no nervous systems that we can identify with confidence. Their structure was simple, often just 2 layers of tissue, an outer epithelium and an inner, largely acellular filling. Some paleontologists describe them as diploblastic, meaning they possessed only 2 primary tissue layers, unlike the triploblastic design of most modern animals, which includes a middle layer that gives rise to muscles and internal organs. One of the most iconic Ediacaran organisms is Charnia, a frond-like form that grew in branching fractal patterns. Charnia was sessile, rooted to the seafloor, and its structure suggests it lived by absorbing dissolved organic matter directly from the surrounding water, a process called osmotrophy. Other Ediacaran organisms may have relied on symbiotic relationships with chemosynthetic bacteria, microbes that convert chemical energy into biological energy. These bacteria thrived on the surface of microbial mats, vast sheets of bacterial colonies that covered much of the ocean floor during this time. The Ediacaran biota, it seems, were not just living on the seafloor, but were integrated into it, dependent on the stable, undisturbed substrate that the mats provided. The microbial mats themselves were crucial to the Ediacaran ecosystem. These mats, composed primarily of cyanobacteria and other microorganisms, formed dense, cohesive layers that stabilized the sediment. They were the foundation of the food web, if such a term even applies to so simple an ecosystem. The mats captured nutrients, hosted symbiotic bacteria, and provided a firm surface for sessile organisms to anchor to. Without the mats, the Ediacaran biota could not have existed in the forms they did. They were, in a very real sense, creatures of the Matground, the stable, mat-covered seafloor that defined their world. This dependence on stability made the Ediacaran biota inherently vulnerable. They were adapted to an environment without disturbance, without predation, without competition in any aggressive sense. Their bodies reflect this. They were soft, lacking any protective structures. They did not move, so they had no need for muscles or the metabolic infrastructure to power them. They did not actively consume food, so they had no need for digestive systems. They were, in many ways, living surfaces, maximizing contact with the water around them, relying on passive diffusion to meet their metabolic needs. The simplicity of their design tells us something important about the environment they inhabited. The Ediacaran oceans were low in free oxygen compared to modern seas, though not entirely anoxic. Oxygen levels had risen during the Neoproterozoic oxygenation event, which began around 850 million years ago, but they had not yet reached the stable, elevated levels necessary to support highly active, energy-intensive life. The Ediacaran biota were adapted to this low-oxygen world. Their passive lifestyle, their reliance on diffusion rather than active circulation, their lack of complex internal organs, all of these features suggest organisms living at the metabolic limit of what was chemically possible at the time. The Ediacaran period was not entirely devoid of change. Over its 35 million year span, the biota diversified, and toward the end of the period, some organisms began to show features that hinted at greater complexity. There are trace fossils, marks left by organisms moving across or through the sediment. though these are rare and simple compared to what would come later. Some paleontologists interpret certain late Ediacaran forms as showing early signs of bilateral symmetry or rudimentary segmentation, but these are hints, not transformations. The fundamental character of the Ediacaran world remained passive, stable, and quiet. Then, at the boundary with the Cambrian, the Ediacaran biota largely disappeared. Not all at once and not entirely, a few forms may have persisted into the early Cambrian, but as a coherent ecosystem, the Ediacaran world came to an end. The reasons for this extinction are tied directly to the rise of the Cambrian fauna. As we will see in later chapters, the new organisms did not simply coexist with the old. They actively dismantled the ecological foundation the Ediacaran biota depended on. Burrowing organisms churned the seafloor, breaking apart the microbial mats. Predators consumed soft-bodied prey. The stable, undisturbed Matground gave way to a dynamic, bioturbated Mixground, and the Ediacaran organisms, so exquisitely adapted to stillness, could not survive the disruption. The Ediacaran period stands as a monument to a different way of being alive, a way that proved ultimately unsustainable, once the conditions that supported it changed. It was a world without urgency, without the constant pressure of predation, without the arms race that would come to define animal life. In that sense, it was a world unlike any that has existed since. Understanding this world, its structure, its limitations, its eventual collapse, is essential to understanding why the Cambrian Explosion happened, when and how it did. The shift from passive to active, from stillness to motion, required more than new organisms. It required a fundamental re-engineering of the planet itself. The seafloor of the Ediacaran period was not bare rock or shifting sand. It was covered, in many places quite thoroughly, by dense mats of microorganisms. These microbial mats, composed primarily of cyanobacteria, along with other bacterial species, formed coherent, carpet-like layers that stabilized the sediment and created a firm surface across the ocean bottom. This Matground ecosystem, as scientists call it, was the ecological foundation for nearly all complex life during the Ediacaran. Without the mats, the biota that lived on them would not have existed in the forms they did. Microbial mats are ancient. They predate complex multicellular life by billions of years. In the Precambrian, before animals appeared, these mats were ubiquitous, covering vast stretches of shallow marine environments. The mats formed through the growth and accumulation of photosynthetic bacteria, which captured energy from sunlight and converted it into organic matter. Over time, the bacteria secreted sticky substances that bound sediment particles together, creating stable, cohesive layers. These layers could be quite thick, several centimeters in some cases, and remarkably durable. They resisted erosion and provided a stable substrate in environments where loose sediment would otherwise have been disturbed by currents. For the Ediacaran organisms, the mats offered multiple advantages. First, they provided a firm surface for attachment. Many Ediacaran forms, such as the frond-like Charnia, were sessile, rooted in place. In a world of soft sediment, anchoring securely would have been difficult without the cohesive structure the mats provided. The mats acted as a kind of biological bedrock, allowing organisms to grow upright into the water column, where they could maximize their surface area for nutrient absorption. Second, the mats were a source of nutrition. The bacteria within the mats were metabolically active, breaking down organic matter and releasing dissolved nutrients into the surrounding water. Some Ediacaran organisms likely absorbed these nutrients directly, using osmotrophy, the passive uptake of dissolved organic compounds through their outer surface. Others may have hosted symbiotic bacteria within their tissues, bacteria that performed chemosynthesis, converting chemical energy from the sediment into usable biological energy. In either case, the mats were central to the Ediacaran food web, providing the base layer of energy that supported the entire ecosystem. Third, and perhaps most importantly, the mats created an environment of stability. Unlike modern seafloors, which are constantly churned by burrowing organisms, the Ediacaran seafloor was largely undisturbed. The microbial mats sealed the sediment surface, preventing it from being easily disrupted. This stability was essential for the Ediacaran biota, whose soft, simple bodies were adapted to a world without sudden changes, without predators or aggressive competitors. The mats enforced a kind of ecological calm, a stillness that allowed passive life strategies to succeed. The Matground ecosystem was not unique to the Ediacaran. Similar microbial mats still exist today, though they are much rarer, restricted to environments where grazing and burrowing organisms are absent, such as hypersaline lagoons or certain deep-sea settings. Modern mats give us a glimpse of what the pre-Cambrian ocean floor might have looked like. Vast expanses of slimy, cohesive bacterial layers, green or brown depending on the species present, hosting a limited community of organisms adapted to slow, passive existence. But the Ediacaran Matground was more extensive and more dominant than any modern analogue. It was the default state of the ocean floor, not an exception, and this dominance shaped the evolutionary trajectory of early complex life. Organisms evolved to exploit the resources the mats provided, to anchor to their surface, to absorb the nutrients they released. In doing so, they became dependent on the mat's persistence. When the mats were destroyed, as they would be during the Cambrian, the organisms that relied on them faced extinction. The destruction of the Matground was not a sudden catastrophe. It was a process, driven by the evolution of new life forms with new behaviors. Mobile organisms, particularly those capable of burrowing into sediment, began to appear in the latest Ediacaran and early Cambrian. These organisms did not respect the integrity of the mats. They churned through the sediment, breaking apart the cohesive bacterial layers, mixing oxygenated water into deeper sediment layers, and creating a fundamentally different substrate. This process, called bioturbation, transformed the seafloor from a stable matground to a dynamic mixground, a churned, oxygenated, constantly disturbed environment. The fossil record documents this transformation. In late Ediacaran sediments, the surfaces of rock layers often show preserved mat textures, delicate wrinkles and patterns left by the bacterial films. By the early Cambrian, these textures become rare and then disappear entirely. In their place, we find trace fossils, burrows and tracks left by mobile organisms moving through the sediment. The transition is stark. Within a geologically brief interval, the character of the seafloor changed completely. For the Ediacaran biota, this change was catastrophic. They had evolved in a world where the substrate was stable, where food was passively available, where there were no predators and no need for defense or escape. The new Cambrian organisms, with their active lifestyles and their ability to burrow, dismantled that world. The mats, once ubiquitous, became fragmented and then largely disappeared from most marine environments. Without the mats, the foundation of the Ediacaran ecosystem was gone. This is one of the most striking examples of ecosystem engineering in Earth's history. The Cambrian organisms did not simply outcompete the Ediacaran biota. They actively destroyed the habitat those organisms required. It was not just biological replacement, but ecological transformation. The rules of life changed, and the old organisms, so perfectly adapted to the old rules, could not adapt to the new ones. The Matground ecosystem, then, is not just a detail of Ediacaran ecology. It is central to understanding why the Cambrian explosion happened the way it did. The rise of mobile, burrowing, active organisms was both a cause and a consequence of the explosion. These new behaviors required new body plans, new metabolic strategies, new sources of energy. And once they emerged, they reshaped the physical environment in ways that made the previous world uninhabitable. The stillness of the Ediacaran gave way to the turbulence of the Cambrian, and the change was permanent. Oxygen is so fundamental to animal life that it is easy to take for granted. Every breath we take, every movement we make, depends on a steady supply of free oxygen molecules. Our cells use oxygen to extract energy from food through a process called aerobic respiration, a metabolic pathway that is far more efficient than the anaerobic alternatives used by many bacteria. Without oxygen, large, active, complex organisms simply cannot exist. This dependence is not incidental. It is a hard biochemical constraint, one that shaped the entire history of complex life on Earth. For most of Earth's history, free oxygen was scarce. The planet's early atmosphere was essentially anoxic, composed primarily of nitrogen, carbon dioxide, methane, and trace amounts of other gases, but almost no free oxygen. Life emerged in this oxygen-poor world, and for billions of years it remained microbial. Bacteria and archaea dominated, many of them anaerobic, capable of metabolizing energy without oxygen. Some, like the cyanobacteria, began producing oxygen as a byproduct of photosynthesis, but for a long time this oxygen did not accumulate. It reacted with dissolved iron in the oceans and with volcanic gases, and was removed from the system as quickly as it was produced. Then, around 2,400,000,000 years ago, something changed. Oxygen began to accumulate in the atmosphere in significant quantities, an event known as the Great Oxidation Event. This was one of the most profound transformations in Earth's history. It was also, from the perspective of many existing organisms, a catastrophe. Oxygen is chemically reactive, capable of damaging cellular machinery. For anaerobic life forms, the rise of oxygen was toxic. Many lineages went extinct or retreated to anoxic refuges, environments like deep sediments or stagnant water where oxygen could not reach them. But for other lineages, oxygen represented an opportunity. Aerobic respiration, the metabolic process that uses oxygen to break down organic molecules, yields far more energy than anaerobic pathways. An organism that could tolerate oxygen and use it for metabolism would have access to a vastly more efficient energy source. This advantage set the stage for the eventual evolution of complex life, but it did not happen immediately. Even after the Great Oxidation Event, oxygen levels remained relatively low compared to modern values. It would take over 1 billion more years for oxygen to rise to concentrations that could support large, active animals. The second major rise in oxygen occurred during the Neoproterozoic era, beginning around 850 million years ago. This event, called the Neoproterozoic Oxygenation Event, saw a sustained increase in atmospheric and oceanic oxygen levels. The causes of this increase are still debated, but they likely involve changes in the carbon cycle, increased burial of organic matter in sediments, which prevented it from consuming oxygen during decomposition, and possibly tectonic activity that altered weathering rates and nutrient availability. Whatever the specific mechanisms, the result was clear. By the late Neoproterozoic, oxygen levels had risen to a point where large, multicellular organisms could survive. But survival is not the same as thriving. The Ediacaran biota, which emerged during this time, were adapted to low oxygen conditions. Their passive lifestyles, their reliance on diffusion for nutrient uptake, their lack of active circulation, all of these traits suggest organisms living close to the metabolic minimum. They could exist in the oxygen levels available, but they could not do much more than exist. Active movement, rapid growth, sustained muscular activity, all of these require higher oxygen concentrations than the Ediacaran world provided. The threshold for complex active animal life is not precisely known, but estimates suggest that atmospheric oxygen needed to reach at least 10 to 15% of present-day levels before large, mobile animals could evolve. Below this threshold, the metabolic costs of maintaining active tissues, particularly muscle, become prohibitively expensive. Oxygen is needed not only for energy production, but also for the synthesis of collagen, a structural protein that forms the basis of connective tissues in animals. Collagen production is an oxygen-intensive process. Without sufficient oxygen, organisms cannot build the robust, flexible tissues needed for complex body structures. The fossil record confirms that oxygen levels reached this threshold by the beginning of the Cambrian. The sudden appearance of large, mobile, predatory organisms that required high metabolic rates to power their lifestyles, indicates that the atmospheric and oceanic oxygen concentrations had stabilized at levels capable of supporting such life. This is not to say that oxygen levels were uniform everywhere. The deep oceans in particular remained largely anoxic during the early Cambrian, with sulfidic or iron-rich waters in many regions. But the shallow seas where most early Cambrian life is found had sufficient oxygen to meet the demands of active organisms. Interestingly, the fact that deep oceans remained anoxic during this time suggests that early animal lineages evolved a degree of metabolic flexibility. Many modern animals retain the ability to survive in low oxygen conditions, at least temporarily, using anaerobic metabolic pathways. This capability, called facultative anaerobiosis, likely reflects the evolutionary history of animals. which first appeared in a world where oxygen was available but not abundant. As oxygen levels rose and stabilized, organisms that could take full advantage of aerobic metabolism gained a decisive edge, but the ancestral tolerance for low oxygen conditions persisted. The rise of oxygen, then, was a permissive factor for the Cambrian explosion. It did not cause the explosion directly, but it removed a fair, fundamental constraint. Without sufficient oxygen, the complex, active bodies that define Cambrian life could not have evolved. Once oxygen reached the necessary threshold, a vast range of new biological possibilities opened up. Organisms could grow larger, move faster, sustain higher metabolic rates, and build more complex tissues. Evolution had the fuel it needed to experiment with new body plans. But fuel alone is not enough. Building complex bodies also requires raw materials, the chemical building blocks for skeletons, shells, and other structural features. The rise of oxygen coincided with another crucial geochemical shift, a massive influx of essential nutrients into the oceans, driven by dramatic changes in Earth's climate and weathering patterns. It is to this second piece of the puzzle that we now turn. Oxygen's role in the Cambrian explosion extends beyond simply providing energy for metabolism. It is also essential for the construction of complex tissues themselves. At the molecular level, the assembly of animal bodies depends on a protein called collagen, and collagen synthesis is fundamentally an oxygen-dependent process. Without sufficient free oxygen in the environment, organisms cannot produce collagen in the quantities needed to build large, structurally sophisticated bodies. This constraint represents a hard biochemical threshold, one that had to be crossed before the Cambrian diversification could occur. Collagen is the most abundant protein in animal bodies. It forms the structural framework for connective tissues, providing strength and flexibility to skin, tendons, ligaments, blood vessel walls, and the extracellular matrix that holds cells together. In animals with skeletons, collagen serves as the organic scaffold onto which minerals are deposited, creating bones and shells. It is, in a very real sense, the material from which complex bodies are built. Without collagen, the intricate architectures that define animal life would be impossible. The synthesis of collagen requires a specific biochemical step that can only occur in the presence of oxygen. The process involves enzymes called prolyl hydroxylases, which modify the amino acid proline after it has been incorporated into the collagen protein chain. This modification, called hydroxylation, is essential for collagen molecules to fold properly and assemble into the strong, rope-like fibers that give the protein its structural properties. The hydroxylation reaction requires molecular oxygen as a substrate. In environments with insufficient oxygen, the reaction cannot proceed efficiently, and functional collagen cannot be produced in adequate amounts. This biochemical requirement creates a direct link between atmospheric oxygen levels and the potential for morphological complexity. As long as oxygen remained below a critical threshold, organisms were constrained in the types of bodies they could build. They could be small, relying on diffusion for oxygen transport and simple tissues for structure. They could be soft, lacking the rigid frameworks that collagen enables. But they could not be large, complex, and structurally robust simultaneously. The Ediacaran biota, with their simple, often gelatinous bodies and lack of complex internal organs, reflect these constraints. When oxygen levels rose during the Neoproterozoic oxygenation event, the biochemical barrier to collagen production was gradually removed. By the late Neoproterozoic and early Cambrian, oxygen concentrations had reached levels where prolyl hydroxylases could function efficiently, allowing organisms to synthesize collagen and build the complex tissues that define modern animals. This chemical permission was a prerequisite for the morphological innovations of the Cambrian. Without it, the genetic and ecological factors that drove diversification would have had no physical substrate to work with. The importance of collagen extends beyond soft tissues. Many of the most iconic features of Cambrian life, mineralized shells and skeletons, depend on collagen as their organic foundation. Biomineralization, the process by which organisms deposit calcium, carbonate or calcium phosphate to form hard structures, requires an organic matrix onto which minerals can crystallize. Collagen provides this matrix. In bone, for example, collagen fibers are arranged in precise patterns, and mineral crystals grow along these fibers, creating a composite material that is both strong and flexible. Without collagen, mineralized skeletons as we know them could not exist. The fossil record reflects this dependence. The earliest evidence of mineralized hard parts appears at the very beginning of the Cambrian period, coinciding with the time when oxygen levels had stabilized at sufficient concentrations for efficient collagen synthesis. Before this point, organisms were almost entirely soft-bodied. After this point, mineralized structures became widespread across multiple unrelated lineages. This simultaneous appearance of skeletonization in diverse groups suggests a shared environmental trigger, most plausibly the crossing of the oxygen threshold necessary for collagen production and, by extension, biomineralization. It is worth noting that not all structural proteins require oxygen for synthesis. Chitin, for example, which forms the exoskeletons of arthropods and is found in fungal cell walls, can be produced under low oxygen conditions. However, chitin alone cannot provide the range of structural solutions that collagen enables. Collagen's unique mechanical properties, its ability to form flexible yet strong fibers, its role in wound healing and tissue repair, make it an irreplaceable component of complex animal bodies. The evolution of collagen-based tissues was a necessary step in the transition from simple to complex multicellular life. The oxygen threshold for collagen synthesis is estimated to be around 10 to 15% of present atmospheric levels, roughly the same threshold required for sustained aerobic metabolism in large organisms. This convergence is not coincidental. Both constraints reflect the same underlying reality. Complex animal life requires a certain minimum concentration of free oxygen to function. Below this level, the biochemical machinery of animal bodies cannot operate efficiently. Above it, a vast range of new possibilities opens up. The late Neoproterozoic and early Cambrian, then, represent the moment when Earth's atmosphere crossed this critical threshold and stayed there. Previous fluctuations in oxygen levels may have occurred, brief rises followed by declines, but the late Neoproterozoic oxygenation was sustained. It provided a stable chemical foundation upon which evolution could build. Organisms that evolved the genetic and developmental systems to produce collagen could now exploit that capability fully, constructing bodies with internal skeletons, protective shells, jointed appendages, and all the other structural features that define complex animals. This chemical permission was necessary but not sufficient. Crossing the oxygen threshold made complexity possible, but it did not make it inevitable. For that, additional factors were needed, specifically the availability of raw materials for building hard structures and the ecological pressures that made such structures advantageous. The stage was set, but the performance required more than just an empty stage. It required props, scenery, and actors, the geological delivery of essential nutrients, the genetic innovations that allowed rapid morphological change, and the ecological interactions that drove organisms to evolve ever more sophisticated adaptations. The collagen threshold, then, is one piece of a larger puzzle. It represents a hard biochemical constraint that had to be overcome before the Cambrian explosion could occur. But it also represents a liberation. Once crossed, this threshold opened up entirely new regions of morphological space, allowing evolution to explore body plans and ecological strategies that had been chemically impossible in earlier eras. The rise of oxygen was not just an environmental change. It was an enabling event, one that fundamentally expanded what life could be. To understand the geochemical conditions that enabled the Cambrian explosion, we must look back to a series of catastrophic climate events that gripped the planet during the Neoproterozoic era. Between roughly 750 million and 580 million years ago, Earth experienced at least two, possibly more episodes of extreme glaciation so severe that ice may have covered the entire planet, from poles to equator. These events, known collectively as Snowball Earth glaciations, were among the most dramatic climate shifts in Earth's history. They were also, paradoxically, essential preparation for the biological revolution that followed. The evidence for Snowball Earth comes from glacial deposits found in rocks that formed at tropical latitudes, regions that today, and presumably in the Neoproterozoic, were too warm for ice sheets. These deposits, called tillites, consist of unsorted sediment and rocks that were transported and deposited by glaciers. Finding them in what were equatorial regions suggests that ice extended far beyond the poles, potentially covering the entire ocean surface. Additional evidence comes from geochemical signatures in rocks, particularly the presence of certain iron minerals that form only in the absence of oxygen, suggesting that the oceans became stratified and largely anoxic beneath the ice. The causes of Snowball Earth glaciations are still debated, but they likely involved a runaway feedback mechanism related to the planet's reflectivity, or albedo. Ice reflects sunlight back into space, cooling the planet further. If ice sheets expanded beyond a certain threshold, the increased reflectivity would have triggered additional cooling, causing more ice formation, which in turn reflected more sunlight. Once this feedback loop began, it may have been difficult to stop, leading to global glaciation. Escaping from a Snowball Earth state required a mechanism powerful enough to overcome the high albedo of an ice-covered planet. The most widely accepted explanation involves volcanic outgassing. Even during a global glaciation, volcanic activity would have continued, releasing carbon dioxide into the atmosphere. Normally, carbon dioxide is removed from the atmosphere through weathering of rocks and uptake by organisms. But during a Snowball Earth, these processes would have been suppressed. Ice covered the continents, preventing weathering. Life, if it survived at all, would have been concentrated in small refuges, unable to consume carbon dioxide at normal rates. As a result, carbon dioxide would have accumulated in the atmosphere over millions of years, gradually building up a greenhouse effect strong enough to melt the ice. The thaw from a Snowball Earth would have been dramatic. As the ice melted, the planet's albedo decreased, allowing more sunlight to be absorbed, which accelerated warming. The transition from a frozen world to a greenhouse world may have occurred relatively rapidly, geologically speaking, within a few tens of thousands to hundreds of thousands of years. The resulting climate would have been hot, humid, and characterized by intense weathering of exposed continental rock. This is where the connection to the Cambrian Explosion becomes clear. The extreme greenhouse conditions following Snowball Earth glaciations drove a pulse of continental weathering far more intense than anything occurring under normal climatic conditions. As temperatures soared and rainfall increased, chemical weathering of rocks accelerated dramatically. Rivers carved through landscapes, carrying dissolved minerals into the oceans at unprecedented rates. Studies of early Cambrian sediments suggest that weathering rates for certain minerals may have been 23 times higher than baseline levels, an extraordinary influx that fundamentally altered ocean chemistry. The minerals delivered by this weathering pulse were not random. They included two elements essential for building complex life: phosphorus and calcium. Phosphorus, typically bound in rocks as phosphate minerals, is a limiting nutrient in many marine ecosystems. It is required for all life, forming the backbone of DNA and RNA molecules, and playing a critical role in energy transfer within cells. In the modern ocean, phosphorus availability often controls biological productivity. When phosphorus is abundant, life blooms. When it is scarce, productivity declines. Calcium, in contrast, is needed specifically for biomineralization. Organisms that build shells or skeletons from calcium, carbonate or calcium phosphate, require a sufficient supply of dissolved calcium ions in seawater. During normal conditions, calcium is present in seawater, but its concentration fluctuates based on weathering rates, volcanic activity, and biological uptake. The post-Snowball weathering pulse delivered enormous quantities of calcium into the oceans, supersaturating the water and making it chemically favorable for organisms to extract calcium and deposit it as solid mineral structures. The timing is significant. The last major Snowball Earth glaciation, known as the Marinoan glaciation, ended around 635 million years ago. This was followed by intense greenhouse warming and the associated weathering pulse. The Ediacaran period began shortly after, around 575 million years ago, and the Cambrian Explosion followed roughly 35 million years later. The interval between the end of Snowball Earth and the beginning of the Cambrian represents a period of geochemical recovery and reorganization, during which the oceans accumulated the nutrients and building materials necessary for complex life. It is important to recognize that the Snowball Earth events were not beneficial in any intentional sense. They were catastrophic for any life that existed at the time. The glaciations likely drove many lineages to extinction, constraining life to small refuges where liquid water persisted, perhaps around volcanic vents or in thin meltwater layers beneath the ice. But by resetting the planet's surface chemistry, by locking up continental weathering during the glaciation, and then releasing it in a massive pulse during the thaw, Snowball Earth created the geochemical conditions that would later enable complexity. This is a recurring theme in Earth's history: destruction as preparation. The Snowball Earth glaciations were not designed to enable the Cambrian Explosion. They were simply extreme climate events driven by the planet's internal dynamics and its orbital relationship with the Sun. But their consequences, the mobilization of continental nutrients and their delivery to the oceans, created an environment chemically rich enough to support the construction of mineralized skeletons and the metabolic demands of active, complex organisms. Without Snowball Earth, the Cambrian Explosion might not have occurred when it did, or in the form it took. The weathering pulse provided the raw materials. The rising oxygen levels provided the energy and the biochemical capacity for collagen synthesis. Together, these geochemical preconditions transformed the ocean from a nutrient-poor, low-energy environment into one capable of supporting rapid diversification and widespread biomineralization. The stage was not just set, it was stocked with everything evolution would need to build a new world. The escape from Snowball Earth glaciations set in motion a chain of geochemical events that would reshape the ocean's chemistry for millions of years. As the ice retreated and the planet swung into an intense greenhouse phase, the exposed continents became sites of extraordinary erosion. Rainfall intensified, rivers swelled, the mechanical and chemical breakdown of rock accelerated to rates far exceeding normal geological conditions. This weathering pulse, as scientists call it, was not a brief event, but a sustained process that delivered massive quantities of dissolved minerals into the global ocean, fundamentally altering the chemical environment in which life existed. Weathering is the process by which rocks break down into smaller particles and dissolve into water. It occurs through both physical mechanisms, such as freeze-thaw cycles and abrasion, and chemical reactions, particularly the interaction of water and carbon dioxide with rock minerals. Under normal conditions, weathering proceeds at relatively steady rates, controlled by climate, topography, and the composition of exposed rock. But following a Snowball Earth event, conditions were far from normal. The planet's climate was hot and humid, driven by the buildup of atmospheric carbon dioxide during the glaciation. Rainfall was likely torrential. The landscape, freshly scoured by glaciers, presented vast expanses of newly exposed rock, ready to be broken down. The result was an erosional catastrophe, geologically speaking. Sediment and dissolved minerals poured into the oceans at rates that may have been 1 to 2 orders of magnitude higher than modern baseline levels. Studies of sedimentary rocks from the late Neoproterozoic and early Cambrian provide evidence for this influx. Certain geochemical proxies, measurements of isotope ratios and trace element concentrations suggest that the flux of continental material into the ocean spiked dramatically during this interval. One study estimated that weathering rates for phosphorus-bearing minerals were potentially 23 times higher than expected under normal conditions. Phosphorus is the key limiting nutrient in most marine ecosystems. Unlike nitrogen, which can be fixed from the atmosphere by certain bacteria, phosphorus has no gaseous phase. It enters the ocean only through weathering of continental rocks. Once in the water, it is rapidly taken up by organisms, incorporated into biological tissues, and either recycled or buried in sediments. Under typical conditions, phosphorus availability is low, constraining biological productivity. But during the post-Snowball weathering pulse, the influx of phosphorus was enormous, removing this constraint and allowing marine life to proliferate at unprecedented rates. Calcium followed a similar trajectory. As silicate minerals in continental rocks broke down, they released calcium ions into solution. These ions were carried by rivers into the ocean, where they accumulated. Calcium is essential for organisms that build mineralized structures. Shells and skeletons are composed primarily of calcium carbonate or calcium phosphate, materials that require both calcium and carbonate or phosphate ions. The weathering pulse provided calcium in abundance, supersaturating the seawater and making biomineralization energetically favorable. The concept of saturation is important here. Seawater can hold dissolved minerals up to a certain concentration, beyond which the minerals begin to precipitate out of solution, forming solid crystals. During the post-Snowball weathering pulse, the concentration of calcium and phosphate in seawater likely approached or exceeded saturation levels in many regions. Under these conditions, the energetic cost of extracting these ions from seawater and incorporating them into shells or skeletons would have been minimal. Biomineralization, which under different conditions might be prohibitively expensive, became not just possible, but relatively easy. This chemical permissiveness explains one of the most striking features of the early Cambrian fossil record: the simultaneous appearance of mineralized hard parts in multiple unrelated lineages. Arthropods, mollusks, brachiopods, echinoderms and other groups all evolved shells, skeletons or protective plates within a narrow window of geological time. This convergent evolution was not driven solely by ecological pressures, though predation certainly played a role. It was also enabled by the chemical environment. The ocean was saturated with the building materials, and organisms that could exploit those materials gained a significant advantage. The ecological consequences of the weathering pulse extended beyond biomineralization. The influx of phosphorus fueled a dramatic increase in biological productivity. Primary producers, organisms like algae and cyanobacteria that convert sunlight into organic matter, bloomed in response to the nutrient availability. This increase in productivity would have rippled up the food web, providing energy for larger, more complex consumers. The Cambrian Explosion was not just a diversification of body plans. It was also an expansion of biomass, a fundamental increase in the amount of living tissue the ocean could support. There is also evidence that the weathering pulse contributed to the stabilization of oxygen levels. As productivity increased, more organic matter was produced. Some of this organic matter sank to the seafloor and was buried in sediments before it could decompose. When organic matter is buried, the oxygen that would have been consumed during its decomposition remains in the atmosphere and ocean. This process, the burial of organic carbon, is one of the primary mechanisms by which oxygen accumulates over geological time. The post-Snowball weathering pulse, by driving a surge in productivity and organic carbon burial, may have helped push oxygen levels over the threshold necessary for complex animal life. The timing of the weathering pulse aligns closely with the emergence of the Ediacaran biota, and later, the Cambrian Explosion. The pulse began shortly after the last major Snowball Earth glaciation ended, around 635 million years ago. The Ediacaran period began approximately 60 million years later, and the Cambrian Explosion followed another 35 million years after that. This sequence suggests a causal chain: glaciation, thaw, weathering pulse, nutrient accumulation, biological diversification, each step built on the previous one, creating conditions that made the next step possible. It is also worth noting that the weathering pulse was not instantaneous. It unfolded over millions of years, gradually altering ocean chemistry. This gradual change allowed ecosystems to adjust, gave evolution time to experiment with new strategies, and ensured that the chemical environment remained favorable for extended periods. The Cambrian Explosion was not a response to a sudden environmental shock. It was the culmination of a long period of geochemical preparation, during which the planet slowly assembled the conditions necessary for complexity. The weathering pulse, then, represents the second major geochemical precondition for the Cambrian Explosion. The first was the rise of oxygen, which provided energy and enabled collagen synthesis. The second was the delivery of raw materials, phosphorus for productivity, and calcium for skeletonization. Together, these two conditions created an ocean environment chemically primed for the rapid evolution and diversification of complex life. But chemistry alone was not enough. Evolution also needed the biological tools to take advantage of these conditions, the genetic and developmental systems that could generate morphological novelty quickly. It is to these biological innovations that we now turn. There is a moment in the history of any ecosystem when a threshold is crossed, when what was merely possible becomes practically inevitable. For the early Cambrian oceans, that moment came when the geochemical conditions the availability of oxygen, the saturation of seawater with essential minerals, aligned to permit widespread biomineralization. Chemistry had always governed what organisms could build, but now it actively encouraged construction. The energetic barriers that had previously constrained skeleton and shell formation were reduced or eliminated, and the selective advantages of possessing hard parts became overwhelming. Biomineralization is the process by which organisms produce mineralized structures, typically by precipitating calcium carbonate or calcium phosphate from surrounding fluids. It is energetically expensive. Cells must actively transport ions across membranes, concentrate them in specific locations, and control the crystallization process to produce structures with the desired mechanical properties. In an environment where calcium and phosphate are scarce, or where the chemical conditions make precipitation difficult, the costs of biomineralization can outweigh the benefits. But when the ocean is supersaturated with these minerals, when the ions are abundant and the chemistry is favorable, the cost decreases dramatically. This is precisely what happened in the early Cambrian. The post-Snowball weathering pulse had delivered enormous quantities of calcium and phosphate into the oceans, raising their concentrations to levels that made biomineralization chemically straightforward. The saturation state of seawater with respect to calcium carbonate, a measure of how easily the mineral will precipitate, was likely higher than at any point in the preceding hundreds of millions of years. Under these conditions, organisms that evolved the cellular machinery to direct mineralization could produce shells and skeletons with relatively little metabolic investment. The advantages of possessing hard parts were manifold. Shells provided protection against predation, a critical benefit in a world where the first multicellular predators were emerging. Skeletons offered structural support, allowing organisms to grow larger and maintain more complex body shapes without collapsing under their own weight. Mineralized structures could also serve as anchors, helping organisms resist being dislodged by currents, or as tools, forming the basis for jaws, claws, and grinding plates used in feeding. In an environment where predation pressure was increasing and where competition for resources was intensifying, the ability to build protective or functional hard parts conferred a decisive survival advantage. What is remarkable about the early Cambrian is not just that biomineralization occurred, but that it occurred independently in so many different lineages within such a short time. Arthropods evolved external skeletons made of calcium-strengthened chitin and later calcite. Mollusks developed shells of calcium carbonate. Brachiopods, organisms that superficially resemble bivalves but belong to an entirely different phylum, also evolved calcium carbonate shells. Echinoderms, the group that includes modern starfish and sea urchins, built skeletons of calcite plates. Even sponges, among the simplest of multicellular animals, began producing mineralized spicules. This convergent evolution across unrelated lineages strongly suggests a common environmental driver. The simultaneous appearance of biomineralization is difficult to explain through ecological or genetic factors alone. Predation pressure, while real, does not explain why such diverse organisms adopted mineralized structures at the same time. The answer lies in the chemistry. The ocean had reached a state where biomineralization was not just advantageous, but chemically permissible in a way it had not been before. Evolution responded to this permission by exploring the possibilities it offered. The fossil record documents this transition with remarkable clarity. In the latest Precambrian, just before the Cambrian boundary, the fossil record is dominated by soft-bodied impressions, organisms with no mineralized structures. Then, at the very beginning of the Cambrian, mineralized fossils appear. Small shelly fossils, fragments of shells, plates and tubes, become abundant in sediments dating to the earliest Cambrian. These structures are often simple, lacking the complexity of later shells, but they represent the first widespread use of biomineralization in the animal kingdom. As the Cambrian progressed, the sophistication of mineralized structures increased. Early shells were often thin and irregular, but later forms developed layered structures, growth lines, ornamentation, and interlocking plates. Organisms learned, in an evolutionary sense, to control the mineralization process more precisely, producing structures optimized for specific functions. The innovation of biomineralization, once established, became a platform for further diversification. One of the most striking early examples of biomineralization is the archaeocyaths, a group of sponge-like organisms that built the first carbonate reefs. Archaeocyaths constructed cup-shaped skeletons of calcium carbonate, cementing themselves to the seafloor and growing in dense colonies. These reefs provided habitat for other organisms, creating complex three-dimensional structures that increased ecological diversity. The existence of archaeocyath reefs in the early Cambrian demonstrates that biomineralization was not limited to individual protection or support. It also enabled ecosystem engineering, the construction of new habitats that supported entire communities. The chemical permissiveness of the early Cambrian Ocean did not last forever. Over time, as organisms consumed calcium and phosphate to build their structures, and as those structures were buried in sediments, the concentration of these minerals in seawater decreased. The ocean's saturation state declined. Biomineralization became, once again, more energetically costly. But by that point, the selective advantage of possessing hard parts was so great that organisms continued to invest in them, even at higher metabolic cost. The innovation had become entrenched. This entrenchment is one of the reasons the Cambrian explosion had such lasting consequences. Once mineralized structures became widespread, they fundamentally altered the nature of ecological interactions. Predators evolved stronger jaws and more efficient feeding mechanisms to deal with armored prey. Prey evolved thicker shells and more elaborate defenses. The arms race, once begun, created its own momentum, driving further innovation even as the environmental conditions that initially enabled biomineralization changed. The chemical conditions of the early Cambrian, then, represent a unique window in Earth's history. For a geologically brief period, the oceans were saturated with the materials needed for construction, oxygen was abundant, and the ecological incentives for building complex bodies were mounting. Evolution seized this opportunity, and the result was the rapid diversification we call the Cambrian Explosion. Chemistry did not determine what forms life would take, but it determined what forms were possible. And in that moment, what was possible was extraordinary. Among the many innovations that define the Cambrian Explosion, one stands out as perhaps the most consequential: the proliferation of organisms with bilateral symmetry. Bilaterians, animals whose bodies can be divided into mirror-image left and right halves, represent a fundamental departure from earlier body plans. This seemingly simple organizational principle, the arrangement of body parts along a central axis, enabled a suite of adaptive features that transformed how organisms interacted with their environment. Bilaterality was not just a structural choice, it was a commitment to a particular way of life, one based on directed motion, active consumption, and continuous engagement with a three-dimensional world. Before bilaterians became dominant, most multicellular organisms exhibited radial symmetry or no clear symmetry at all. Radial organisms, like modern jellyfish or sea anemones, are organized around a central point, with body parts radiating outward. This design works well for passive or slow-moving creatures that interact with their environment from all directions simultaneously. A jellyfish, drifting with currents and capturing prey with tentacles arranged in a circle, does not need to distinguish between front and back, left and right. Its lifestyle is fundamentally non-directional. Bilaterality, in contrast, imposes directionality. A bilaterian organism has a defined front and back, a head end and a tail end. This organization is optimized for movement through the environment along a consistent axis. The head end leads, encountering new stimuli first. The tail end follows. This simple asymmetry has profound consequences. It favors the concentration of sensory organs and neural processing at the front of the organism, leading to cephalization, the development of a distinct head region. It also enables the streamlining of body shape, reducing drag and improving efficiency during locomotion. The internal architecture of bilaterians reflects this directional organization. Most possess a one-way digestive system, often described as a tube within a tube design. Food enters through a mouth at the front, passes through a specialized digestive tract where nutrients are extracted, and exits through an anus at the rear. This arrangement is far more efficient than the two-way systems seen in simpler organisms, which must use the same opening for both ingestion and egestion. A one-way gut allows for regional specialization, Different sections of the digestive tract can be optimized for different tasks: grinding, chemical breakdown, nutrient absorption, waste compaction. This specialization supports continuous feeding and more complete extraction of energy from food. The origin of bilaterality is still debated. The earliest bilaterians likely evolved in the late Neoproterozoic, before the Cambrian began. Trace fossils, burrows and tracks left by mobile organisms appear in sediments dating to the latest Ediacaran, suggesting that some animals had already adopted a bilaterian body plan and were using it to move through sediment in search of food. But these early forms were likely small, simple, and rare. It was during the Cambrian that bilaterians diversified explosively, giving rise to the vast majority of animal phyla that exist today. The success of bilaterians in the Cambrian can be attributed to the ecological opportunities that directed motion created. A mobile organism can seek out food rather than waiting for it to arrive. It can avoid unfavorable conditions and actively select habitats. It can escape predators or conversely pursue prey. This active lifestyle requires energy, which is why the rise of bilaterians coincided with the availability of sufficient oxygen. But the advantages it conferred were so significant that once bilaterality emerged, it quickly became the dominant body plan in marine ecosystems. Cephalization, the concentration of nervous tissue and sensory organs at the head end, is a direct consequence of directed motion. If an organism is moving forward, it is advantageous for sensory organs to be positioned at the front, where they encounter stimuli first. Eyes, antennae, chemoreceptors, organs for detecting light, chemicals, touch and vibration all cluster around the head in bilaterians. This sensory concentration is paired with neural centralization, the development of a brain or brain-like ganglion that processes sensory information and coordinates responses. The bilaterian nervous system is organized to support rapid decision-making, allowing organisms to react quickly to threats and opportunities. The fossil record from the Cambrian shows a remarkable diversity of bilaterian forms. Arthropods, with their segmented bodies and jointed appendages, are among the most abundant and morphologically varied. Annelids, segmented worms, appear in trace fossils and, occasionally, in sites with exceptional preservation. Mollusks, with their muscular foot and protective shell, are also present. Chordates, the group that includes vertebrates, are represented by small fish-like forms, such as those found in the Chengjiang biota. Each of these groups exhibits bilaterality, and each uses this body plan in slightly different ways, adapted to different ecological niches. Bilaterality also enabled the evolution of complex behaviors. Mobile organisms can exhibit directed responses to stimuli, moving toward food or mates, fleeing from predators, navigating complex environments. These behaviors require coordination between sensory input, neural processing, and muscular output, all features that are more developed in bilaterians than in other animal groups. The capacity for complex behavior, in turn, opened up new ecological roles. Bilaterians could be predators, active hunters that pursue and capture prey. They could be scavengers, seeking out dead organic matter. They could be burrowers, engineers that reshape their environment. The commitment to bilaterality was also a commitment to a higher metabolic rate. Directed motion requires sustained muscular activity, which demands a continuous supply of energy. This is why the rise of bilaterians is so closely linked to the availability of oxygen. Without sufficient oxygen, the metabolic machinery needed to power an active, mobile lifestyle cannot function. But once oxygen levels rose, bilaterians were uniquely positioned to exploit this resource, using it to fuel the energetically expensive processes of movement, sensation, and neural activity. The Bilaterian Blueprint, then, is more than an anatomical curiosity. It is a solution to a specific ecological challenge: how to be an active, mobile consumer in a complex, competitive environment. The success of this solution is evident in the fossil record and in the modern world. The vast majority of animal species today are bilaterians. From insects to fish, from worms to mammals, the basic body plan, bilateral symmetry, cephalization, one-way gut, remains remarkably consistent. It was established during the Cambrian, refined and elaborated over the subsequent half-billion years, but never fundamentally replaced. The Cambrian Explosion was, in many ways, the triumph of bilaterality. The Bilaterian body plan, with its front-to-back orientation and mirror symmetry, necessitated innovations in internal organization that would prove foundational to all subsequent animal complexity. Two features in particular, the concentration of sensory and neural structures in a head region and the development of a through-gut digestive system, transformed how organisms interacted with their environment and processed the resources they encountered. These were not incidental refinements, but fundamental architectural solutions to the challenges of active, mobile life. Cephalization, the development of a distinct head, emerged as a direct response to directional movement. When an organism moves consistently in one direction, the leading edge encounters environmental stimuli first. It makes functional sense to concentrate sensory organs at this leading edge, where they can detect food, threats, or obstacles before the rest of the body arrives. In early bilaterians, this meant clustering light-sensitive cells, chemoreceptors for detecting dissolved chemicals, and mechanoreceptors for sensing touch and vibration near the front of the organism. Sensing stimuli, however, is only useful if an organism can respond appropriately. This requirement drove the evolution of concentrated neural tissue, clusters of nerve cells that could integrate sensory information and coordinate behavioral responses. In the simplest bilaterians, this neural concentration took the form of cerebral ganglia, small clusters of neurons near the major sensory organs. In more complex forms, these ganglia enlarged and became more sophisticated, eventually giving rise to recognizable brains. The fossil record from the Cambrian, particularly from exceptional preservation sites like Chengjiang, occasionally preserved evidence of neural tissue, dark stains in the head region that represent decay products of concentrated nervous systems. The advantages of cephalization were substantial. A centralized processing center allowed for faster, more coordinated responses than would be possible with a diffuse nerve net, the type of nervous system seen in radially symmetric animals like jellyfish. When a Cambrian arthropod detected a predator with its eyes, the visual information traveled a short distance to nearby ganglia, which could immediately trigger escape behavior, activating specific leg muscles in a coordinated sequence. This speed and precision were essential in an environment where predation was becoming a dominant ecological force. The head region also became the location of the mouth, the entry point for food. This placement was logical. The front of the organism encountered food first, so positioning the mouth there allowed for immediate consumption. But the mouth itself underwent evolutionary refinement during the Cambrian. In the simplest bilaterians, it may have been little more than an opening, a gap in the body wall through which food could enter. As predation and specialized, feeding strategies evolved, mouths became more complex, developing structures for grasping, tearing, or filtering food. The arthropods, in particular, show remarkable diversity in mouth structures during the Cambrian. Some possessed mandibles, hard mouth parts for crushing and grinding. Others had filter-feeding appendages for straining small particles from the water. Still others developed raptorial appendages, modified limbs positioned near the mouth that could capture and manipulate prey. This morphological diversity in feeding structures reflects the ecological diversity of early Cambrian environments and demonstrates how the basic bilaterian body plan could be modified to exploit different food sources. The evolution of a through gut, a digestive system with a mouth at one end and an anus at the other, was equally transformative. This design, sometimes called a complete gut or alimentary canal, contrasts with the blind gut systems seen in simpler organisms, where a single opening serves both for ingestion and digestion. The through gut is fundamentally more efficient. Food enters the mouth, travels in one direction through a tube, where digestion and absorption occur in sequence, and waste exits through the anus. There is no backflow, no mixing of incoming food with outgoing waste. This directional flow allows for regional specialization within the digestive tract. Different sections can be optimized for different functions. The anterior portion might serve for mechanical breakdown, grinding food into smaller particles. The middle sections might be specialized for chemical digestion, secreting enzymes that break down complex molecules into absorbable nutrients. The posterior portions could focus on water absorption and waste compaction. This division of labor increases the overall efficiency of digestion, allowing organisms to extract more energy from each meal. The through gut also enabled continuous feeding. An organism with a blind gut must wait for digestion to complete and waste to be expelled before consuming more food. An organism with a through gut can ingest food at one end while waste is being expelled from the other. This continuous processing supports higher metabolic rates and allows for more sustained activity. For active predators or grazers, this was a significant advantage. Evidence for through guts in Cambrian organisms comes both from fossilized gut traces, visible as dark lines running through the body in exceptionally preserved specimens, and from coprolites, fossilized feces, which demonstrate that organisms were processing food and expelling waste as discrete pellets. The presence of coprolites in early Cambrian sediments is itself significant. It indicates not only that through guts had evolved, but that organisms were consuming and processing substantial quantities of food, reflecting higher metabolic rates than earlier, passive organisms. The combination of cephalization and a through gut created an integrated feeding machine. Sensory organs in the head detected food. The brain processed this information and directed the organism toward the food source. The mouth captured and ingested the food. The gut processed it efficiently, extracting maximum nutrition, and the organism used this energy to continue moving, searching for more food. This positive feedback loop, successful feeding enabling more activity, which enabled more successful feeding, drove the evolutionary success of bilaterians during the Cambrian. The internal organization of bilaterians also included the development of a body cavity, or coelom, a fluid-filled space between the gut and the outer body wall. This cavity served multiple functions. It provided space for internal organs to develop and move independently of the body wall. It acted as a hydrostatic skeleton in soft-bodied forms, allowing for more sophisticated locomotion through controlled muscle contractions. And it facilitated the circulation of nutrients and gases, compensating somewhat for the lack of specialized circulatory systems in many early forms. The fossil record shows that by the early Cambrian, these features, heads with concentrated sensory and neural structures, through guts with regional specialization, body cavities supporting internal organs, were already well established in multiple lineages. The speed with which these innovations appeared and spread suggests that they were strongly favored by selection. In an environment where resources were abundant but competition was intense, where predators were emerging and prey needed to become more efficient at finding food or avoiding being eaten, the bilaterian body plan with its integrated head and gut systems provided decisive advantages. These anatomical innovations were not independent developments, but interrelated components of a functional whole. The head directed the organism toward food. The gut processed that food efficiently. The energy extracted powered the muscles that moved the organism. Each component enabled and reinforced the others, creating an integrated system that was greater than the sum of its parts. This integration is characteristic of bilaterian animals and distinguishes them fundamentally from the passive, decentralized organisms that preceded them. The Cambrian explosion was not simply the appearance of new body plans. It was the assembly of functional systems, integrated solutions to the challenges of active life in a complex competitive environment. Heads and guts were central to these solutions. They enabled organisms to sense their world, find resources, process them efficiently, and use the resulting energy to continue the cycle. Once this system was established, it proved remarkably durable. The same basic organization, head with brain and sensory organs, through gut with regional specialization, persists in bilaterians today, from the simplest flatworms to the most complex vertebrates. The morphological diversity that appeared during the Cambrian explosion, dozens of fundamentally different body plans arising within 20 million years, presents a puzzle. Natural selection typically operates gradually, through the accumulation of small genetic changes over many generations. Mutations arise randomly, and those that improve survival or reproduction become more common. But this process is slow. Building a complex new structure, A jointed leg, a compound eye, a segmented body requires numerous coordinated changes. How could so much innovation occur so quickly? The answer lies not in the genes themselves, but in how those genes are regulated. The genome of an organism contains the instructions for building proteins, the molecular machines that perform most biological functions. But simply having genes is not enough. The organism must also control when and where those genes are expressed, which genes are active in which cells at which times during development. This control is exercised by regulatory networks, systems of genes that act as switches, turning other genes on or off in response to developmental signals. Among the most important regulatory genes are the Hox genes, a family of master control genes that determine the identity of body segments along the head-to-tail axis. Hox genes were discovered through studies of fruit fly development, where mutations in these genes cause dramatic transformations, legs growing where antennae should be, or extra wings appearing in the wrong body segments. These genes do not code for legs or wings directly. Instead, they specify which segment of the body a particular group of cells belongs to, and this specification determines what structures those cells will eventually build. Hox genes are ancient. They are found in all bilaterians, from simple worms to complex vertebrates, and the basic set of Hox genes was likely present in the common ancestor of all bilaterian animals. This means that Hox genes existed before the Cambrian Explosion. The explosion did not require the invention of new genes, but rather new ways of using existing genes. This distinction is crucial. Adding entirely new genes to a genome is slow, requiring duplication events and subsequent divergence. Modifying regulatory networks, changing how existing genes are deployed, can happen much faster. The hypothesis that has emerged to explain Cambrian diversification is sometimes called the regulatory evolution or developmental systems theory. It proposes that the rapid morphological innovation of the Cambrian was driven primarily by changes in gene regulation rather than changes in the genes themselves. Small modifications to the regulatory regions of genes, the DNA sequences that control when and where a gene is expressed, can have large effects on the organism's body plan. These modifications can create new structures by expressing genes in novel locations or at novel times, or they can modify existing structures by altering the intensity or duration of gene expression. Consider the arthropod appendage, a limb composed of segments that can be modified into legs, antennae, mouth parts, gills, or reproductive structures. All of these diverse forms are built using the same basic genetic toolkit. The difference lies in which genes are expressed during the development of each appendage. A gene that promotes hardening might be highly expressed in a mandible, but suppressed in a gill. A gene that promotes elongation might be active in an antenna, but inactive in a leg. By mixing and matching gene expression patterns, evolution can generate a vast array of appendage forms without inventing new genes for each one. This flexibility is what made the Cambrian Explosion possible. Once the basic bilaterian body plan and the core regulatory networks, particularly the Hox system, were in place, evolution could rapidly explore morphological space. A mutation in a regulatory region might cause a leg-like appendage to develop in a new body segment, creating a novel structure that could be selected for if it improved survival. Another mutation might alter the size or shape of that appendage, refining its function. Through iterative cycles of regulatory change and selection, complex new structures could assemble relatively quickly. The fossil record supports this interpretation. The Chengjiang biota, dating from the early Cambrian, already shows a stunning diversity of arthropod body plans. Some forms have many similar segments, each bearing a similar limb. Others show pronounced differentiation, with specialized appendages in different body regions. This variability suggests that the regulatory systems controlling segment identity and appendage development were actively evolving, being tinkered with by natural selection to produce forms adapted to different ecological niches. Gene duplication also played a role, though likely a supporting one. When a gene is duplicated, creating two copies in the genome, one copy can maintain the original function while the other is free to evolve new functions. Hox genes themselves have undergone duplication events during animal evolution, expanding from a small ancestral set to the larger, more complex clusters seen in modern animals. These duplications provided raw material for innovation, additional switches that could be co-opted for new purposes. But the crucial factor was not the number of switches, but the rewiring of the networks that controlled them. There is also evidence that non-Hox regulatory genes underwent significant diversification during the Cambrian. Genes involved in cell signaling, genes that control how cells communicate with each other during development, show signs of expansion and modification during this period. These signaling systems are essential for coordinating the growth and differentiation of complex structures. Changes in signaling pathways could alter the size, shape, or position of body parts, contributing to the morphological diversity of Cambrian fauna. The concept of modularity is important here. Biological structures are often modular, composed of semi-independent units that can be modified or rearranged without disrupting the entire organism. A segmented body is modular. Each segment can develop somewhat independently. An appendage is modular. Different parts, segments, joints, terminal structures can be modified separately. This modularity, which is built into the developmental programs controlled by regulatory networks, allows evolution to tinker with one part of an organism without breaking everything else. It accelerates innovation by reducing constraints. The Cambrian Explosion, then, was not a sudden mutation bonanza, a rapid accumulation of random genetic changes. It was a period of intensive regulatory evolution, a rewiring of the developmental software that translates genetic information into physical form. The hardware, the genes themselves, was largely in place before the explosion began. What changed was the software, the control networks that determined how those genes were used. This shift from genetic to regulatory innovation explains how so much morphological diversity could appear so quickly. It also explains why the Cambrian was unique. Once the major body plans were established, once the regulatory networks were optimized for particular lifestyles, further innovation became more constrained. Evolution could still modify and elaborate, adding new species, new adaptations, new ecological strategies. But inventing entirely new body plans became increasingly difficult, because doing so would require dismantling established developmental programs that were now essential to producing viable organisms. The Cambrian was a window of regulatory flexibility, a period when developmental systems were still malleable enough to be radically reorganized. That window eventually closed, not because evolution stopped, but because the systems it had built became too integrated, too essential to be fundamentally redesigned. The emergence of predation during the Cambrian period represents one of the most consequential ecological innovations in Earth's history. For the first time, organisms actively pursued, captured, and consumed other organisms. This shift from passive coexistence to active antagonism transformed the selective pressures operating on all life forms. Predation introduced urgency into evolution. It created an arms race that has continued, unabated, for over 500 million years. And it fundamentally reshaped marine ecosystems, favoring organisms with the sensory, locomotory, and defensive capabilities to survive in a world where being eaten was a constant possibility. Before the Cambrian, feeding strategies were predominantly passive or detritivorous. Ediacaran organisms likely absorbed dissolved nutrients from the water, or relied on symbiotic bacteria. Early bilaterians may have grazed on microbial mats or consumed small particles of organic matter. These strategies did not require complex sensory systems, high metabolic rates, or sophisticated locomotion. The appearance of predators changed this calculus entirely. Suddenly, every organism was either a potential predator or potential prey, or both. Survival required new adaptations. The earliest evidence for predation comes from the pre-Cambrian-Cambrian boundary, in the form of boreholes drilled through the shells of early shelly fossils. These holes, precisely circular, and often positioned at specific locations on the shell, suggest deliberate attempts to access the soft tissue inside. The organisms responsible for these holes are unknown, but their existence demonstrates that by the very beginning of the Cambrian, some animals had evolved the ability to breach protective structures and consume the organisms within. As the Cambrian progressed, predators became larger, faster, and more specialized. The most iconic example is Anomalocaris, a genus of large, free-swimming predators found in Middle Cambrian deposits. Anomalocaris could reach lengths of up to 1 meter, enormous by the standards of early Cambrian life. Its body was streamlined for swimming, with lateral lobes that undulated to generate thrust. It possessed large, compound eyes, among the most sophisticated visual organs of the Cambrian, capable of detecting movement and forming images, and it had a pair of specialized appendages at the front of its body, spiny, jointed structures that could grasp and manipulate prey. The mouth of Anomalocaris was unlike anything seen in earlier organisms. It consisted of a ring of plates arranged in a circular pattern, capable of contracting to crush or slice food. Fossilized gut contents, preserved in rare specimens, show that Anomalocaris consumed hard-shelled organisms, indicating that its feeding apparatus was strong enough to crack protective armor. Anomalocaris was not just a predator, but an apex predator, occupying the top of the food web in Middle Cambrian ecosystems. The existence of apex predators implies a structured food web with multiple trophic levels. At the base were primary producers, organisms like algae that converted sunlight into organic matter. Above them were small grazers and filter feeders, consuming microorganisms or detritus. Above those were secondary consumers, smaller predators or scavengers, and at the top were animals like Anomalocaris, predators with no natural enemies. This ecological stratification is characteristic of modern ecosystems, but was a novel feature of the Cambrian world. Predation exerted intense selective pressure on prey organisms. Those with effective defenses, thick shells, rapid escape responses, cryptic coloration, were more likely to survive and reproduce. Those without such defenses were more likely to be consumed before reproducing. This created a feedback loop. As prey evolved better defenses, predators faced selective pressure to evolve better offensive capabilities. As predators became more effective, prey faced pressure to enhance their defenses further. This reciprocal selection, each side driving the evolution of the other, is known as a co-evolutionary arms race. The fossil record documents this arms race. Early Cambrian shells are often thin and simple, providing minimal protection. By the Middle Cambrian, shells had become thicker, more robust, and often elaborately ornamented with spines or ridges that made them harder to grasp or penetrate. Some organisms evolved the ability to withdraw completely into their shells, sealing the opening with a protective plate. Others adopted burrowing as a defensive strategy, hiding in sediment where predators could not easily reach them. Still others evolved rapid locomotion, using jet propulsion or powerful swimming strokes to escape threats. Sensory systems also underwent rapid evolution in response to predation. Eyes, in particular, became more sophisticated. The ability to detect a predator at a distance and initiate escape before it arrived was a powerful selective advantage. The Cambrian saw the evolution of complex compound eyes in arthropods, camera-type eyes in some mollusks and vertebrates, and various other photoreceptor structures. Vision became a dominant sense, shaping behavior and ecology in ways that persist to the present. Predation also drove the evolution of behavioral complexity. Organisms that could learn to recognize predators, remember safe refugees, or coordinate group defenses had higher survival rates. This required more sophisticated nervous systems, with enhanced capacity for sensory integration, memory formation, and decision-making. The elaboration of neural structures during the Cambrian, evidenced by larger cerebral ganglia in fossil specimens, may have been driven in part by the cognitive demands of predator-prey interactions. The ecological impact of predation extended beyond direct predator-prey relationships. Predators can control the abundance of prey populations, preventing any single species from dominating an ecosystem. This top-down control can increase diversity by preventing competitive exclusion, allowing multiple prey species to coexist. Predators also influence prey behavior, causing prey to spend more time in refugees or less productive habitats, which can have cascading effects throughout the ecosystem. The appearance of predators during the Cambrian may have been a key factor in the maintenance of the high diversity that characterizes post-Cambrian marine ecosystems. It is also worth noting that predation created ecological opportunities. Scavengers, organisms that feed on dead predators or their leftovers, could exploit resources that would otherwise go unused. Parasites, organisms that live on or in other organisms without immediately killing them, represent another strategy enabled by the existence of complex animal bodies. The diversification of ecological strategies during the Cambrian was not limited to predators and their prey, but extended to all the roles that arise in a complex multi-level food web. The emergence of predation, then, was not simply the addition of a new feeding strategy. It was a fundamental reorganization of ecological interactions. It introduced dynamism, urgency, and relentless selective pressure into marine ecosystems. It drove the evolution of defensive and offensive innovations, from thicker shells to sharper claws, from better vision to faster swimming, and it created the ecological structure, the multi-level food webs, the predator-prey dynamics that have characterized animal life ever since. The Cambrian arms race, once initiated, never ended. It continues today, 500 million years later, shaping the evolution of every organism in every ecosystem. The widespread evolution of mineralized shells and skeletons during the early Cambrian has long been recognized as one of the defining features of the period. These hard structures appear suddenly in the fossil record, across multiple unrelated lineages, within a narrow window of geological time. While the geochemical conditions that made biomineralization possible, specifically the saturation of seawater with calcium and phosphate, were discussed earlier, chemistry alone does not fully explain why so many organisms adopted this innovation simultaneously. The answer lies in ecology, in the urgent necessity of defense in a world where predation had become a dominant force. Shells and skeletons are energetically expensive to produce. Organisms must extract minerals from seawater, concentrate them within cells, transport them to sites of deposition, and control the crystallization process to produce structures with specific mechanical properties. This requires specialized cellular machinery, significant metabolic investment, and often a substantial portion of the organism's total energy budget. In an environment without predation, where the primary challenges are finding food and avoiding unfavorable conditions, the costs of building armor might outweigh the benefits. But in an environment with predators, armor becomes essential. The fossil evidence for predation pressure is direct and unambiguous. Early Cambrian shells, even small ones, often bear bore holes, circular penetrations created by predators attempting to access the soft tissue inside. These holes are not random. They are often positioned at specific mechanically weak points on the shell, suggesting that predators had evolved not just the ability to drill, but also the behavioral sophistication to target vulnerable areas. The presence of these bore holes in some specimens, and their absence in others suggests that predation attempts were not always successful. Thicker shells, better sealed openings, or more robust construction could prevent penetration, conferring a survival advantage. Experimental studies on modern organisms support the hypothesis that shell thickness and robustness respond to predation pressure. When snails are raised in environments with predatory crabs, they develop thicker shells than genetically identical snails raised without predators. The snails detect chemical cues from the crabs and adjust their shell growth accordingly. This phenotypic plasticity, the ability to modify development in response to environmental conditions, likely existed in early shell-bearing organisms as well. Those lineages that could increase shell thickness in response to predation risk would have had higher survival rates, and natural selection would have favored the genetic mechanisms underlying this plasticity. The variety of shell architectures that evolved during the Cambrian reflects different solutions to the problem of defense. Some organisms built thick, robust shells that were difficult to crush or penetrate. Others constructed shells with multiple layers, each layer oriented in a different direction, creating a composite structure that resisted cracking. Still others added spines, ridges, or other protrusions that made the shell harder to grasp or maneuver. Each of these design solutions represents an evolutionary experiment in defense, tested by predation pressure and refined by natural selection. Internal skeletons also provided defensive benefits, though in less obvious ways. A rigid internal framework allows an organism to grow larger without collapsing under its own weight. Larger size can be a defense in itself, placing the organism beyond the size range of most predators. Internal skeletons also enable more powerful locomotion, providing attachment points for muscles that generate stronger, faster movements. An organism that can swim or crawl quickly has a better chance of escaping a predator than one that is slow or sessile. The evolution of hard parts was not instantaneous. Early shells were often thin, incomplete, or poorly mineralized, suggesting that the cellular machinery for biomineralization was still being refined. Some early Cambrian organisms possessed only partial protective covering, small plates or scales rather than complete shells. These intermediate forms indicate that shell evolution proceeded in stages, with each incremental improvement in protection conferring a survival advantage and being favored by selection. Over successive generations, shells became thicker, more complete, and more effective. There is also evidence that some organisms experimented with alternative defensive strategies before or instead of evolving shells. Burrowing, the act of moving into and through sediment, provides protection by removing the organism from the reach of surface-dwelling predators. The Cambrian substrate revolution, which will be explored in greater detail in the next chapter, was driven in part by organisms adopting burrowing as a defensive strategy. Trace fossils from the early Cambrian show increasingly complex burrow structures with multiple openings, vertical shafts, and horizontal galleries, suggesting that burrowing behavior became more sophisticated over time. Mobility itself is a defense. An organism that can detect a predator at a distance and rapidly move away has a higher survival probability than one that must rely solely on passive protection. The evolution of sophisticated sensory organs, particularly eyes, and the neural systems to process visual information and coordinate escape responses were driven in part by the need to detect and evade predators. The close association between the evolution of vision and the evolution of locomotion during the Cambrian is not coincidental. Both were responses to predation pressure. Some organisms adopted cryptic strategies, evolving body shapes or color patterns that allowed them to blend into their surroundings. While color is rarely preserved in fossils, body shape and texture can be, and some Cambrian organisms show morphologies consistent with camouflage. Flattened bodies that could press against the substrate, irregular outlines that mimicked rocks or algae, and surface textures that matched the surrounding sediment are all features that could reduce detection by visually hunting predators. Chemical defenses, though difficult to detect in the fossil record, may also have been employed. Modern organisms use toxins, unpleasant tastes, or noxious secretions to deter predators. If a predator consumes a toxic prey item and survives, it may learn to avoid similar prey in the future. This form of defense, called aposematism, is often coupled with bright warning colors or distinctive patterns. Whether Cambrian organisms employed chemical defenses is unknown, but given their prevalence in modern ecosystems, it is plausible that they evolved during the Cambrian arms race. The rapid diversification of defensive strategies during the Cambrian demonstrates the power of predation as a selective force. Within a geologically brief interval, organisms evolved an extraordinary range of solutions to the problem of not being eaten. Some solutions involve physical barriers, shells and skeletons. Others involve behavior, burrowing, fleeing, hiding. Still others likely involve chemistry, toxins and deterrents. Each solution was tested against the evolving capabilities of predators, and those that worked were retained and refined. The necessity of armor then was not abstract or hypothetical. It was immediate and survival critical. In a world where predators roamed, where boreholes in shells and crushed exoskeletons testified to constant danger, organisms without effective defenses simply did not survive to reproduce. The fossil record is biased toward organisms that successfully defended themselves, those with thick shells, effective escape responses, or protective burrows. Those without such defenses left no fossils because they left no descendants. Predation pressure acted as a filter, shaping the morphology, behavior, and ecology of all Cambrian organisms. The armor we see in Cambrian fossils is not decoration. It is necessity, carved into the bodies of organisms by the relentless pressure of being potential prey. While predation transformed interactions above the seafloor, a quieter, but equally consequential revolution was unfolding within the sediment itself. Mobile organisms, Particularly early bilaterians with a capacity for directed locomotion began to actively burrow into the substrate, moving through the upper layers of sediment in search of food or refuge. This behavior, called bioturbation, fundamentally altered the physical and chemical properties of the seafloor. It disrupted the microbial mats that had dominated for hundreds of millions of years, changed the flow of nutrients and oxygen through sediment layers, and ultimately drove the extinction of the Ediacaran biota, whose entire lifestyle was built on the stability those mats provided. The Ediacaran seafloor was a Matground ecosystem, characterized by dense, cohesive microbial mats that stabilized the sediment surface. These mats formed a seal, a biological barrier that prevented oxygen from penetrating deeply into the sediment and that kept the sediment particles bound together. Organisms lived on top of the mats, anchored to their surface or lying flat against them. The mats were the foundation of the ecosystem, and their stability was essential. Any disruption to the mats would cascade through the entire community. The first burrowers were likely small and simple, making shallow horizontal traces just beneath the sediment surface. Trace fossils from the latest Ediacaran, known as ichnofossils, show simple horizontal burrows and surface trails, evidence of organisms moving through or across the sediment. These early traces are rare and shallow, suggesting that burrowing was not yet a dominant behavior. But by the early Cambrian, trace fossils become more common, more diverse, and more complex. Vertical burrows appear, indicating organisms moving up and down through the sediment. Three-dimensional networks develop, with multiple tunnels intersecting at various depths. The density of traces increases, showing that many organisms were burrowing simultaneously. One of the most significant early Cambrian trace fossils is Treptichnus pedum, a complex burrow system that has been chosen as the defining marker for the start of the Cambrian period. Treptichnus consists of a series of horizontal segments connected by vertical shafts, creating a stair-step pattern. The organism that made this trace, identity unknown, was actively mining the sediment, moving through it in 3 dimensions, likely feeding on organic matter or microorganisms within the sediment layers. The appearance of Treptichnus marks a threshold, the point at which burrowing became sufficiently widespread and intensive to leave a clear signal in the rock record. Bioturbation had multiple effects on the sediment. First, it physically disrupted the microbial mats. As burrowing organisms moved through the upper sediment layers, they broke apart the coherent bacterial films, fragmenting them into isolated patches. This disruption reduced the mat's ability to stabilize sediment, making the seafloor more susceptible to erosion by currents. It also reduced the mat's capacity to host the symbiotic relationships and nutrient cycling that had supported the Ediacaran biota. Second, bioturbation introduced oxygen into deeper sediment layers. When a burrowing organism moves through sediment, it creates a tunnel that allows oxygenated water from above to penetrate downward. This oxygenation changes the chemistry of the sediment, enabling aerobic bacteria to colonize deeper layers and altering the cycling of nutrients. In anoxic sediments, many minerals remain in reduced forms that are chemically unavailable to organisms. Oxygenation oxidizes these minerals, making them accessible. This process, called bioirrigation, increased the overall productivity of the sediment ecosystem. Third, bioturbation mixed the sediment vertically, bringing nutrients from deeper layers to the surface and transporting fresh organic matter from the surface downward. This mixing, combined with oxygenation, created a more dynamic, heterogeneous sediment environment. The stable, layered structure of the Ediacaran Matground gave way to a churned, homogenized Mixground. This new substrate was more favorable for organisms adapted to active lifestyles, organisms that could burrow, feed within the sediment, and tolerate the variability that bioturbation created. For the Ediacaran biota, the transition from Matground to Mixground was catastrophic. Their sessile, passive lifestyle depended on the stability of the microbial mat substrate. As the mats were broken apart and the sediment became unstable, the Ediacaran organisms lost their essential habitat. They could not adapt to the new conditions because their entire body plan was optimized for a stable, undisturbed environment. They lacked the mobility to relocate, the defensive structures to protect against disturbance, and the physiological flexibility to tolerate the oxygenated churned sediment. Within a geologically brief period, the Ediacaran biota largely disappeared from the fossil record, replaced by the active mobile fauna of the Cambrian. The transformation of the seafloor was not uniform or instantaneous. In some environments, particularly deep or anoxic settings, microbial mats persisted longer. In shallow, well-oxygenated coastal areas, bioturbation occurred earlier and more intensely. The fossil record shows a mosaic pattern, with different environments transitioning at different times. But the overall trend was clear. By the Middle Cambrian, the Mixground substrate had become the norm in most marine environments, and the Matground ecosystem was restricted to marginal habitats, where burrowing organisms were rare or absent. The Cambrian substrate revolution, as this transformation is called, is one of the most profound examples of ecosystem engineering in Earth's history. Ecosystem engineering occurs when organisms modify their physical environment in ways that affect other species. Beavers building dams, trees creating forest canopies, and corals constructing reefs are all examples of ecosystem engineering. The Cambrian burrowers engineered the substrate itself, transforming it from a stable mat-covered surface into a dynamic, bioturbated zone. This engineering was not intentional. The organisms were simply feeding or seeking refuge. But the cumulative effect of countless individual burrowing events reshaped the seafloor, and, in doing so, drove the extinction of an entire biota and enabled the establishment of a new ecological order. The feedback loops involved in the substrate revolution were self-reinforcing. As more organisms adopted burrowing, the sediment became more disturbed, which made it less favorable for mat-building bacteria and more favorable for organisms adapted to churned conditions. This positive feedback accelerated the transition, pushing ecosystems past a tipping point beyond which return to the previous state became impossible. The Matground-Mixground transition was a one-way shift, a permanent change in the structure of marine sediment ecosystems. The ecological consequences extended beyond the Ediacaran extinction. The Mixground substrate created new ecological niches. Organisms could now feed within the sediment, grazing on bacteria, or consuming organic particles mixed into the sediment by bioturbation. Predators could hunt within the sediment, pursuing burrowing prey through their tunnels. The three-dimensional exploitation of the substrate increased habitat complexity and supported higher biodiversity. The Cambrian seafloor was not just more active than the Ediacaran seafloor, it was more ecologically rich, with more ways for organisms to make a living. The Cambrian substrate revolution demonstrates a fundamental principle of evolutionary ecology. Organisms shape their environments, and those environments, in turn, shape subsequent evolution. The burrowing behavior that destroyed the Ediacaran Matground created the conditions for further diversification of burrowing organisms, which created further disturbance, which created more niches. The process was cumulative, each generation of organisms inheriting an environment modified by the previous generation and modifying it further for the next. This niche construction, as it is called, is a powerful evolutionary force, one that played a central role in the Cambrian Explosion and continues to operate in ecosystems today. The transformation of the seafloor from a stable, mat-dominated surface to a churned, oxygen-rich substrate represents one of the most enduring legacies of the Cambrian Explosion. This shift, which unfolded over several million years during the earliest Cambrian, was not merely a change in sediment texture, but a fundamental reorganization of marine ecology. The Matground ecosystem, which had persisted for hundreds of millions of years, was permanently replaced by the Mixground system that characterizes most marine sediments today. Understanding this transition requires examining not just what changed, but why the change was irreversible. The Matground ecosystem was built on stability. Microbial mats grew slowly, layer upon layer, creating a cohesive surface that resisted physical disturbance. These mats were self-maintaining. The bacteria within them secreted sticky extracellular substances that bound sediment particles together. The mat surface deflected water currents, reducing erosion, and the mat's dense structure prevented larger organisms from easily penetrating it. As long as the mat remained intact, it provided a stable platform for sessile organisms and a firm substrate for crawling forms. The ecosystem was in equilibrium, maintained by the mutual reinforcement of its components. Bioturbation disrupted this equilibrium. Each burrowing organism that pushed through the sediment broke apart a small section of the microbial mat. Individually, these disruptions might have been minor, potentially repairable, as bacteria recolonized the disturbed area. But as burrowing organisms became more abundant during the early Cambrian, the rate of disturbance exceeded the mat's capacity for regeneration. The mats became fragmented, reduced from continuous carpets to isolated patches, and once fragmented, they became vulnerable to further degradation. The introduction of oxygen into deeper sediment layers altered the chemical balance that had maintained mat dominance. In anoxic conditions, certain metabolic pathways that benefit mat-forming bacteria are favored. Sulfate-reducing bacteria, for example, thrive in anoxic sediments and contribute to mat formation. When oxygen penetrates the sediment through burrow walls, aerobic bacteria colonize these oxygenated zones, outcompeting the anaerobic forms. The microbial community composition shifts, and the mat-forming species decline. This chemical change is self-reinforcing. More oxygen leads to fewer mat-forming bacteria, which leads to less cohesive sediment, which allows more burrowing, which introduces more oxygen. The mixing of sediment layers had nutritional consequences. In a stable matground, organic matter settled on the surface and was either consumed by surface-dwelling organisms or decomposed by bacteria within the mat. Nutrients cycled primarily at the sediment-water interface, but in a bioturbated mixground, organic matter is transported downward by burrowing organisms, burial, and active mixing. This creates vertical nutrient gradients within the sediment. Organisms that could access these subsurface nutrients by burrowing or by extending feeding structures into the sediment gained access to resources that were previously locked away. This created selective pressure for deeper burrowing and more sophisticated feeding strategies. The physical properties of the sediment also changed. Matground sediment, bound by microbial films, was firm and cohesive. Walking or crawling across it did not require specialized adaptations. Mixground sediment, constantly churned and loosely packed, was softer and less stable. Organisms living on or in this substrate needed different morphological solutions. Some evolved broader appendages to distribute weight and prevent sinking. Others developed streamlined bodies for easier movement through the sediment. Still others adopted permanent burrowing lifestyles, constructing reinforced tunnels that provided stable microhabitats within the unstable substrate. The irreversibility of the transition stems from multiple factors. First, once burrowing organisms became established, their activities maintained the mixground state. Each generation inherited a bioturbated substrate and continued to bioturbate it. There was no opportunity for mats to regenerate on a large scale because the disturbance was constant. Second, the ecological niches created by the mixground system were filled by organisms adapted to exploit them. These organisms depended on bioturbation for access to food and oxygen. If mat conditions returned, many of these species would disappear, but their abundance and diversity made them collectively capable of preventing mat re-establishment through their own activities. Third, the extinction of the Ediacaran biota removed potential mat stabilizers. Some Ediacaran organisms may have contributed to mat stability through their passive presence, their bodies reducing water flow across the sediment surface, or their metabolic activities supporting mat-forming bacteria. Once these organisms disappeared, eliminated by the substrate transformation they could not survive, the ecosystem lost components that might have helped maintain matground conditions. The new ecosystem, dominated by burrowing and mobile forms, had no functional equivalents. The geographical pattern of the transition provides insight into its mechanisms. Bioturbation appeared first in shallow, well-oxygenated coastal environments where conditions favored active, mobile organisms. These were areas with high nutrient availability, strong currents, and variable conditions that selected for adaptability. In deeper, more stable environments, or in regions with low oxygen levels, matground conditions persisted longer. Some deep-sea environments today still harbour microbial mats. In settings where grazing and burrowing organisms are rare, due to low food availability or harsh chemical conditions. These modern matgrounds are ecological relicts, remnants, of a substrate type that was once globally dominant but is now restricted to marginal habitats. The timescale of the transition varied by location, but globally, the shift from matground dominance to mixground dominance was largely complete by the Middle Cambrian. Trace fossil diversity and abundance increased dramatically during this interval. The thickness of the bioturbated zone, the depth to which organisms actively churned the sediment, also increased, expanding from a few millimeters in the late Ediacaran to several centimeters or more by the Middle Cambrian. This deepening bioturbation zone represents not just more burrowing, but deeper burrowing, indicating that organisms were exploiting progressively deeper sediment layers. The ecological consequences of this transformation were profound and enduring. The mixground substrate increased habitat complexity, providing more niches and supporting higher biodiversity. It enhanced nutrient cycling, making locked-away resources available to organisms. It increased oxygenation of sediments, expanding the volume of seafloor that could support aerobic life. And it created selective pressures that drove further innovation in morphology, behavior, and ecology. The organisms that engineered this transformation were also shaped by it, in a feedback loop that accelerated diversification. The Cambrian substrate revolution was not a single event, but a process, a cascading series of changes driven by the interaction of biological activity and environmental response. It demonstrates that evolution operates not just on organisms, but on entire ecosystems. The seafloor itself evolved during the Cambrian, transformed from a passive, stable surface into an active, dynamic zone of biological activity. This transformation was a necessary foundation for the diversification that followed. Without the mixground substrate, many of the ecological strategies that characterize modern marine life would not be possible. The Cambrian seafloor, churned and oxygenated by the activities of mobile organisms, became the stage on which all subsequent marine evolution would unfold. The understanding of Cambrian diversity depends fundamentally on geological accidents, rare circumstances under which soft tissues that would normally decay within days or weeks instead fossilized, preserving evidence of organisms that would otherwise leave no trace. These exceptional preservation sites, known as fossil Lagerstätten, provide the primary evidence for the true morphological complexity of Cambrian life. Without them, our view of the Cambrian explosion would be dramatically impoverished, limited to the minority of organisms that evolved mineralized hard parts. The Lagerstätten offer windows into ancient ecosystems, snapshots of biological diversity frozen at specific moments in deep time. Most fossilization occurs through a straightforward process. An organism dies, its soft tissues decay rapidly through bacterial action and scavenging, and only the hard parts, shells, bones, teeth, remain to be buried in sediment and gradually mineralized. This process is biased. It preserves only organisms with hard parts and only the hard parts of those organisms. Internal organs, muscles, guts, eyes, nervous systems, gills, and other soft structures decay and disappear. For understanding basic anatomy, diversity of hard-shelled organisms, and general ecological patterns, this normal fossilization is adequate. But for understanding the full complexity of Cambrian life, it is woefully insufficient. Exceptional preservation requires exceptional circumstances. The most important factor is rapid burial, ideally in conditions that exclude oxygen. When an organism is buried quickly in fine-grained sediment, particularly in an environment where oxygen is absent or severely depleted, the bacteria and scavengers that would normally consume soft tissues cannot operate effectively. Decay slows or stops. In some cases, minerals from the surrounding sediment infiltrate the tissues, replacing organic molecules with stable mineral forms, while preserving the original structure. In other cases, organic molecules themselves are preserved as thin carbonaceous films. Either way, the result is a fossil that retains not just the organism's outline, but details of its internal anatomy. The circumstances that produce exceptional preservation are rare. They typically occur in specific depositional settings, often involving rapid mud flows or turbidity currents that bury organisms suddenly, or in stagnant basins where the water column and sediment are anoxic. These settings represent a tiny fraction of total marine environments, which means that Lagerstätten preserve only a small, geographically restricted sample of ancient biodiversity. But they are invaluable samples. They capture organisms that are entirely absent from normal fossil assemblages and reveal anatomical details that transform scientific understanding of evolutionary relationships and ecological complexity. The type of exceptional preservation seen in Cambrian Lagerstätten is often called Burgess, shale-type preservation, named after the most famous of these sites. BST preservation is characterized by the replacement or coating of soft tissues with phyllosilicate minerals, particularly clays, which create two-dimensional or low-relief impressions on rock surfaces. These fossils can preserve extraordinary detail, individual muscle fibers, segments of the digestive tract, the structure of gills, the arrangement of appendages, even in rare cases the outline of neural tissue. Under microscopy, some specimens reveal cellular-level structures. This level of preservation allows paleontologists to reconstruct not just what organisms look like, but how they functioned, how they moved, what they ate, how they sensed their environment. The geological settings that produce BST preservation share common features. Most are associated with deeper water environments, below the storm wave base, where fine-grained sediment accumulates slowly under normal conditions but can be suddenly disrupted by underwater landslides or sediment flows. When such flows occur, they can transport organisms from shallower environments and bury them along with the sediment they carry. The transported organisms, mixed with the sediment in low oxygen conditions, are preserved as the flow settles. The fine grain size of the sediment is crucial. Coarse sediment does not mold around delicate structures, but fine mud can preserve minute details. The discovery of Lagerstätten has revolutionized paleontology repeatedly. Each new site expands the known range of Cambrian diversity, reveals previously unknown body plans, and fills gaps in understanding of evolutionary relationships. The sites are distributed globally, from North America to China to Greenland to Australia, demonstrating that the exceptional preservation conditions that create Lagerstätten occurred in different locations at different times. Each site offers a slightly different window, preserving fauna from different depths, latitudes, and ecological settings. Together they provide a composite picture of Cambrian life far richer than any single site could offer. Despite their importance, Lagerstätten represent a biased sample. They preserve primarily organisms from specific environments, usually deeper water settings where the preservation conditions occur. Organisms from shallow high-energy environments, such as wave-swept coastlines or tidal zones, are underrepresented because rapid burial in anoxic conditions is rare in those settings. This means that a significant portion of Cambrian diversity, organisms that lived in shallow water and lacked hard parts, is likely missing from the fossil record entirely. Paleontologists must always remember that even Lagerstätten, exceptional as they are, do not capture the full picture. The study of Lagerstätten fossils requires specialized techniques, because soft tissue preservation is often subtle, visible only under certain lighting conditions or after careful preparation. Specimens must be examined using multiple methods. Low-angle lighting can reveal surface relief that is invisible under direct illumination. Microscopy allows detailed examination of fine structures. Chemical analysis can identify residual organic molecules or mineral compositions that provide clues about the original tissues. In recent years, techniques like scanning electron microscopy and synchrotron imaging have revealed details invisible to the naked eye, including three-dimensional structure preserved within rocks. The interpretation of Lagerstätten fossils also requires caution. Soft tissues can be deformed during burial and preservation, leading to distorted body shapes. Different tissues preserve differently, creating a taphonomic bias where some structures are overrepresented and others underrepresented. And the assignment of fossils to modern taxonomic groups can be challenging, especially when dealing with extinct body plans that have no living representatives. Paleontologists must carefully distinguish genuine anatomical features from preservational artifacts, a task that requires extensive experience and comparative study. Despite these challenges, Lagerstätten have provided some of the most profound insights into the Cambrian explosion. They have revealed the earliest vertebrates, confirming that our own lineage extends back to the very beginning of animal diversification. They have documented the diversity of arthropod body plans, demonstrating that this group experimented with far more morphological variations than survive today. They have shown that complex ecological interactions, predation, scavenging, symbiosis, were already well established in the early Cambrian. Without Lagerstätten, these insights would be impossible. The windows of exceptional preservation, rare and scattered though they are, illuminate the Cambrian world in ways that normal fossils cannot. Among the sites of exceptional preservation, the Chengjiang biota of southern China holds a position of particular importance. Dating to approximately 520 million years ago, early in the Cambrian period, Chengjiang preserves a diverse assemblage of soft-bodied organisms that provides the earliest detailed snapshot of complex animal life. While the Burgess Shale in Canada is more famous, Chengjiang is older, capturing fauna closer to the beginning of the Cambrian Explosion, and thus offering more direct evidence of the rapidity and nature of early animal diversification. The Chengjiang deposits formed in a marine shelf environment in water depths of perhaps 50 to 200 meters. This was not the deep ocean, but not the shallowest coastal zone either, a middle depth setting where sediment accumulated steadily, and where occasional mudflows or storm events could bury organisms rapidly. The fine-grained mudstones that make up the Chengjiang Formation preserved soft tissues with exceptional fidelity. Individual fossils often show complete body outlines, articulated appendages, digestive systems, gill structures, and other features that would normally decay within hours or days of death. The diversity captured at Chengjiang is remarkable. Over 250 species have been identified from these deposits, representing nearly all major animal phyla. Arthropods dominate the fauna, as they do in most Cambrian assemblages, with forms ranging from small, simple creatures to large, complex predators. But Chengjiang also preserves sponges, cnidarians, pripulid worms, various worm-like forms of uncertain affinity, early echinoderms, brachiopods, and, most significantly, for understanding vertebrate evolution, primitive chordates. The arthropods of Chengjiang display an extraordinary range of body plans. Some possess numerous similar body segments, each bearing a pair of appendages, a design that suggests evolutionary experimentation with modularity. Others show pronounced differentiation among segments, with specialized appendages for different functions: walking, feeding, sensory reception, reproduction. Many Chengjiang arthropods have large, well-developed compound eyes, indicating that vision was already a crucial sense. The presence of diverse feeding strategies, filter feeding, predation, scavenging, is evident from the variety of mouthpart structures preserved. One of the most remarkable aspects of Chengjiang is the preservation of animals that left no shell or skeleton, but possessed complex internal anatomy. Prepulid worms, for example, are found in abundance. These organisms had aversible proboscises, muscular structures that could be extended from the mouth for feeding, often lined with teeth or spines for grasping prey. The fossil specimens show the proboscis structure in detail, revealing how these soft-bodied predators captured and processed food. Without Chengjiang, prepulid diversity in the Cambrian would be almost entirely unknown, as these organisms rarely fossilize in normal conditions. But the most consequential discovery from Chengjiang is the presence of early chordates, organisms that belong to the phylum that includes all vertebrates. The most famous Chengjiang chordate is Haikuichthys, a small fish-like animal approximately 2 to 3 centimeters long. Haikuichthys possessed a notochord, a flexible rod running along the back that provided skeletal support, a defining feature of chordates. It had segmented muscle blocks, visible in the fossils, that allowed for side-to-side swimming motions. It possessed gill slits for respiration and a distinct head region, with possible sensory organs. These features place Haikuichthys firmly within the chordate lineage, making it one of the oldest known vertebrate ancestors. The significance of finding chordates in the early Cambrian cannot be overstated. It confirms that vertebrate lineage, the group to which humans and all other backboned animals belong, extends back to the very beginning of animal diversification. It demonstrates that complex body plans, including features like segmented musculature and specialized sensory structures, evolved rapidly once the environmental and genetic conditions were favorable, and it validates the hypothesis that the Cambrian explosion was truly explosive in terms of morphological innovation, with major body plan diversity appearing within the first 10 to 15 million years of the period. Chengjiang also preserves evidence of complex ecological relationships. Fossilized gut contents show what organisms were eating. Predator-prey size relationships can be inferred from the diversity of body sizes and feeding structures present. The presence of burrowing organisms alongside surface-dwelling forms indicates habitat stratification, and the overall diversity and abundance of fossils suggests that early Cambrian ecosystems were already complex. Multi-level food webs with primary producers, herbivores, predators, and scavengers all present. The age of Chengjiang relative to other Lagerstätten is crucial. The Burgess Shale, while more famous and historically more important to paleontology, dates to the Middle Cambrian, approximately 505 million years ago. By that time, animal diversification was already well advanced. Chengjiang, at 520 million years ago, is 15 million years older, which places it much closer to the onset of the Cambrian explosion. This temporal positioning means that Chengjiang provides more direct evidence of how quickly complex body plans appeared. The presence of sophisticated chordates, diverse arthropods, and complex ecological interactions just 15 million years after the start of the Cambrian confirms that the diversification was front-loaded, occurring rapidly at the beginning of the period, rather than unfolding gradually throughout it. The preservation quality at Chengjiang varies. Some specimens are exquisitely detailed, while others show only outlines or partial structures. This variation reflects differences in burial conditions, decay state at the time of burial, and subsequent geological alteration. But even incomplete specimens contribute to understanding. The sheer abundance of fossils at Chengjiang allows statistical analysis of community composition, body size distributions, and ecological patterns that would be impossible with smaller samples. Chengjiang has also been important for resolving debates about the interpretation of Cambrian fossils. Some organisms found at other sites, known only from partial or poorly preserved specimens, have been better understood through comparison with well-preserved Chengjiang examples. Anatomical details visible in Chengjiang fossils have clarified evolutionary relationships and ecological roles that were previously uncertain. In this sense, Chengjiang serves as a reference standard, a site where preservation is good enough that interpretations can be made with confidence and then applied to fossils from other localities. The discovery and study of Chengjiang is also a reminder of how much remains unknown. The site was first scientifically described in the early 1980s, relatively recently in the history of paleontology. Since then, new species continue to be discovered as more rock is excavated and examined. Each new find expands understanding of early Cambrian diversity and sometimes challenges existing hypotheses. The fossil record is not a completed puzzle but an ongoing discovery, with each new Lagerstätte and each new specimen adding pieces to the picture. Chengjiang provides the earliest detailed testimony of complex animal life, capturing a moment just after the threshold had been crossed, when the genetic, ecological, and environmental conditions had aligned to enable morphological diversity. The organisms preserved there demonstrate that by 520 million years ago, the Cambrian explosion was already producing the fundamental body plans that would define animal life for the next half-billion years. The speed and completeness of this transformation, documented in the rocks of southern China, remain among the most profound puzzles and revelations in the history of life on Earth. The Lagerstätten, for all their importance, represent only a fraction of Cambrian environments. The exceptional preservation conditions that create these sites occur primarily in deeper water, low-energy settings, where fine-grained sediment and anoxic conditions allow soft tissues to fossilize. But much of the ocean's biological activity during the Cambrian, as now, occurred in shallow water environments, coastal zones, tidal flats, sunlit shelves where productivity was highest, and ecological interactions most intense. These shallow environments were characterized by strong currents, abundant oxygen, and active scavenging, conditions that destroy soft tissues rapidly and prevent the formation of Lagerstätten. For decades, understanding of life in these productive shallow zones was limited to the hard-shelled organisms that could fossilize under normal conditions. Recent advances in paleontological techniques have begun to reveal this hidden diversity. One approach involves the extraction and analysis of small carbonaceous fossils, or SCFs, from shallow water sediments. These are microscopic organic remains, fragments of cuticle, tiny shell pieces, jaw elements that are too small to be easily visible but can be recovered through careful chemical treatment of rock samples. By dissolving the mineral matrix of sedimentary rocks in acid, paleontologists can concentrate the organic residues and examine them under high magnification. What emerges from this process is evidence of abundant, diverse life in environments that seemed barren when examined with traditional methods. The SCF assemblages from early Cambrian shallow water deposits are dominated by arthropod remains, particularly fragments of cuticle and feeding structures. This is not surprising. Arthropods were diverse and abundant throughout the Cambrian, and their chitinous exoskeletons, while organic rather than mineral, are more resistant to decay than most soft tissues. But the specific structures preserved in the SCF record reveal details about arthropod ecology and diversity that would otherwise be unknown. Jaw elements, mandibles, and maxillae used for processing food show remarkable variation in size, shape, and ornamentation, indicating a diversity of feeding strategies. Some jaws are robust and heavily toothed, adapted for crushing hard-shelled prey. Others are delicate and finely serrated, suited for filtering or grasping soft-bodied organisms. The presence of diverse jaw morphologies in shallow water SCF assemblages demonstrates that ecological specialization had already progressed far by the early Cambrian. Organisms were not generalists competing for the same resources, but specialists, each adapted to exploit particular food sources or occupy particular niches. This level of functional diversity, comparable to that seen in modern marine ecosystems, indicates that the ecological framework established during the Cambrian explosion was sophisticated from the beginning. It was not a gradual accumulation of complexity, but a rapid assembly of complete functioning ecosystems. Mollusks are also represented in SCF assemblages, primarily through fragments of their radulae, the tooth-bearing feeding structures unique to this group. The radula is a ribbon-like organ covered with rows of chitinous teeth, used to scrape algae from surfaces or rasp flesh from prey. The morphology of radula teeth varies according to diet, with different tooth shapes adapted for different feeding modes. Early Cambrian SCF assemblages preserve multiple distinct radula types, indicating that mollusks had already diversified into multiple ecological roles. Some radulae show simple, file-like teeth suitable for grazing on microbial films. Others display more complex hook-like teeth that suggest predation or scavenging. The significance of finding multiple radula morphologies in early Cambrian sediments extends beyond simply documenting mollusk diversity. It demonstrates that mollusks, which would become one of the most successful animal groups in subsequent geological periods, had already established their fundamental feeding strategies during the Cambrian explosion. The radula, an innovation unique to mollusks, was being deployed in multiple ways, allowing the group to exploit a range of food resources. This early ecological diversification likely contributed to the long-term success of mollusks. The SCF record also captures evidence of organisms that are entirely absent from Lagerstätten. Some microscopic fragments cannot be assigned to known groups, suggesting the existence of organisms with body plans that are poorly understood or that left no representatives in the better-preserved deep-water assemblages. These enigmatic fossils are reminders that Cambrian diversity exceeded what is captured even in exceptional preservation sites. The total biological diversity during the Cambrian was likely substantially higher than current fossil evidence can document. Another approach to revealing hidden diversity involves re-examination of sediments previously thought to be fossil-poor. By applying new imaging techniques, including ultraviolet fluorescence and spectroscopic methods, paleontologists have discovered faint organic traces that were invisible under normal light. These traces, sometimes little more than chemical stains, can preserve the outlines of soft-bodied organisms. While they do not provide the anatomical detail of Lagerstätten fossils, they expand the known geographic and environmental range of Cambrian life, showing that complex organisms inhabited a wider variety of settings than the Lagerstätten alone would suggest. The hidden diversity of shallow Cambrian environments has implications for understanding the explosion's causes and mechanisms. If shallow water ecosystems were already ecologically complete and diverse by the early Cambrian, this suggests that the environmental conditions enabling complexity, oxygen availability, nutrient abundance, geochemical favorability were widespread, not restricted to particular settings. It also suggests that the ecological drivers of diversification, predation pressure, competition for resources, habitat complexity operated across environments, creating selective pressures that were consistent throughout the marine realm. The geographic distribution of SCF assemblages also provides evidence for the global nature of the Cambrian explosion. Early Cambrian shallow water deposits from China, Siberia, Australia, and North America all yield diverse SCF assemblages with broadly similar compositions. While there are regional differences in species composition, the presence of similar functional groups, predatory arthropods, grazing mollusks, various worm-like forms, indicates that the ecological framework was consistent across continents. The Cambrian explosion was not a local phenomenon, but a planetary transformation occurring simultaneously in marine environments worldwide. The study of hidden diversity is ongoing. Each new analytical technique reveals previously undetected fossils. Each new fossil locality expands the known range of Cambrian life. And each discovery refines understanding of how diverse and ecologically complex early Cambrian ecosystems actually were. The traditional view of the Cambrian explosion, based primarily on Lagerstätten and hard-shelled fossils, portrayed a world that was diverse but perhaps not as ecologically sophisticated as modern oceans. The emerging picture from SCF studies and other techniques suggests otherwise. By the early Cambrian, marine ecosystems had already achieved a level of functional complexity and trophic structure comparable to what exists today. The hidden diversity, glimpsed through microscopic fragments and faint chemical traces, testifies to the thoroughness and rapidity of the Cambrian transformation. The Cambrian explosion stands alone in the history of life as a moment when fundamental body plans, the basic architectural templates for animal form, were established. In the half-billion years since, evolution has elaborated on these templates, producing extraordinary species diversity and ecological variety. But it has not invented new templates. The phyla that appeared during the Cambrian remain the phyla that exist today. No fundamentally new body plan, no new way of organizing tissues into a functional animal body has emerged since that brief geological window closed. This uniqueness raises a profound question. Why was the Cambrian non-repeatable? The answer lies in the convergence of conditions that occurred during the late Neoproterozoic and early Cambrian. Multiple factors, geological, geochemical, genetic, and ecological, aligned simultaneously, creating a unique set of circumstances that enabled rapid morphological innovation. Each factor was necessary, but none alone was sufficient. Only when all pieces came together did the explosion occur. And once it occurred, once the major body plans were established, the conditions that enabled their invention could not recur because the world had fundamentally changed. The geochemical conditions were specific to that moment in Earth's history. The post-Snowball Earth-weathering pulse delivered unprecedented quantities of phosphorus and calcium into the oceans, saturating seawater with the materials needed for biomineralization and supporting high biological productivity. Oxygen levels had risen to the threshold necessary for active, large-bodied organisms, but had not yet reached modern concentrations. This particular combination, sufficient oxygen to enable complexity, but an ocean still recovering from Snowball Earth and thus chemically enriched, was a transient state. Within tens of millions of years, as organisms consumed the nutrients and as oxygen levels stabilized, the extreme chemical favorability declined. Later organisms evolved in more moderate chemical conditions. The genetic conditions were also particular to that time. The early bilaterians possessed Hox gene systems and other regulatory networks, but these systems were still relatively flexible. not yet locked into producing specific body plans. The genetic architecture allowed for large-scale reorganization. Regulatory mutations could generate novel morphologies without immediately compromising viability. As body plans became established and as organisms became more complex, this flexibility decreased. Developmental programs became more integrated, more interdependent. Changing one component would disrupt others. The genetic systems became canalized, constrained to produce particular outcomes. Early in bilaterian evolution, there was morphological possibility space that could be explored. Later, that space narrowed. The ecological conditions of the early Cambrian also contributed to the explosion's uniqueness. The oceans were full of unexploited resources and empty niches. There were no established ecosystems to invade, no incumbent species to outcompete. The first organisms to evolve particular adaptations, vision, jointed limbs, protective shells, faced little competition from forms already possessing those features. They could diversify rapidly, filling available niches before others arrived. This ecological opportunity only existed once. After the Cambrian, subsequent diversifications occurred in ecosystems already populated by diverse, well-adapted organisms. New body plans would have to compete with established forms, a much higher barrier. The concept of developmental constraint helps explain why new phyla have not appeared since the Cambrian. As organisms become more complex, their developmental processes become more tightly coordinated. Early development, the stages that establish basic body organization, is particularly constrained. Mutations that alter early development usually produce non-viable organisms because they disrupt the foundation upon which later development builds. Late-stage development, the elaboration of structures already specified in early stages, is more flexible. Evolution can tinker with late stages, modifying the size, shape, or function of structures without catastrophic consequences. But inventing an entirely new body plan requires altering early development, and this is increasingly unlikely to produce viable organisms as developmental systems become more complex and integrated. There is also the matter of ecological entrenchment. Once ecosystems were filled with organisms adapted to particular niches. Those organisms modified their environments in ways that reinforced their own ecological success. Predators created selective pressure for better defenses, which created pressure for better predatory adaptations, locking both sides into an arms race. Organisms that churned the substrate maintained mixground conditions that favored other churning organisms. Reef-building organisms created habitats that supported communities adapted to reef environments. These ecological feedbacks stabilized the existing system, making radical departures less likely to succeed. Subsequent major diversification events in Earth's history illustrate the contrast with the Cambrian. The Great Ordovician biodiversification event, which occurred approximately 50 million years after the Cambrian explosion, saw an enormous increase in species diversity. The number of marine families nearly tripled. Reefs expanded. Ecological complexity increased. But this diversification occurred within existing phyla. It was a radiation of species, not a radiation of body plans. The Ordovician added branches to the evolutionary tree, but the trunk and major limbs had been established during the Cambrian. The same pattern holds for later events. The Mesozoic marine revolution, the Cenozoic diversification of mammals, the radiation of flowering plants, all represent major increases in species diversity and ecological complexity, but none produced fundamentally new higher-level body plans. Evolution was operating in elaboration mode, not invention mode. The architectural possibilities had been explored during the Cambrian, and subsequent evolution worked within the boundaries established then. This does not mean evolution has been static since the Cambrian. The changes have been profound. Flight evolved independently in insects, pterosaurs, birds, and bats. Complex brains developed in cephalopods and vertebrates. Social behavior, parental care, symbiotic relationships, all evolved and diversified. But these innovations occurred within existing phyla, using modifications of existing body plans. No organism has evolved a fundamentally new way of being an animal, a new organizational principle comparable to the difference between a jellyfish and an arthropod, or between a worm and a vertebrate. The Cambrian Explosion was a phase transition in the history of life, a moment when the system crossed a threshold and entered a new stable state. Before the Cambrian, life was simple, microbial, or simple multicellular. After the Cambrian, life was complex, organized into elaborate bodies with specialized tissues and organs. The transition was rapid because multiple enabling factors converged, and it was non-repeatable because once those factors had operated, once body plans were established and ecosystems filled, the conditions that had enabled such rapid innovation no longer existed. Understanding the Cambrian as non-repeatable does not diminish its significance. If anything, it enhances it. The Cambrian established the biological framework for all subsequent animal evolution. Every vertebrate, including humans, inherits a body plan invented during that brief window. Every ecosystem functions according to ecological principles, predator-prey dynamics, trophic structure, niche partitioning, that were established during the Cambrian. The world we inhabit, in its fundamental biological organization, is a Cambrian world, and it has been so for more than 500 million years.